Neem Summit. I request Dr. A.K. Handa, Principal Secretary, ICAR-CAFRI, to chair the session, and I request Dr. Rajarajan, Senior Scientist, ICAR-CAFRI, to chair the session, to co-chair the session, and the speakers for this morning session are Dr. Anamika Bose, Dr. Kumaran, Dr. Gopinath, and Dr. Devendra Kumar. I request all the speakers also to come and occupy the seats on the dais. Thank you very much.
Morning, everyone. So we will start the session, and first is Dr. Anamika Bose. First speaker is Dr. Anamika Bose.
Dr. Anamika Bose is an assistant professor with the Department of Pharmaceutical Technology at Naipur, Punjab. She holds a PhD from Jadavpur University on the field of cancer immunology. She has published many research articles in peer-reviewed journals and guided students also. Now the floor is open to Dr. Anamika Bose. Thank you. It is displayed for you.
Okay.
Okay. And if at all any change is required, you can tell him. He will be watching your movement. You can just say he will change it also.
So I can change. I can't change. No?
You just see. No, there is no chart. No. Only he has to change. Only he has to change. No, pointer is there. But this is a LED pointer. It will not be pointing there. It will not work there. Changing, he will. It's working. It's working. Yeah.
A very good morning to all. It is my pleasure and privilege to be here in this platform, which is celebrating the power of Neem. I wish to thank Dr. Nirmala Kothari and WNO organizers for inviting me, for giving me this opportunity to share our Neem research in this platform in front of this August gathering. So today I'll be talking about a glycoprotein which we isolated from neem leaf glycoprotein, and emerging options to target cancer stem cells to restrict cancer metastasis. Okay. No, I feel comfortable here. So all of you must know that cancer is the second leading cause of death worldwide. The conventional treatment modalities, which include surgery, chemotherapy, and radiotherapy, is mainly able to restrict the primary tumor burden alone or in combinations, but they failed to restrict the cancer recurrence and metastasis. As well as these therapies can create lot of additional toxicities. On the other hand, in recent years, in last decades, we witnessed revolutions in cancer management with the help of immunotherapy, which basically educate or trained our body's own defense cells to target the cancer cells. There are different types of immunotherapies are there, but mainly checkpoint inhibitors are gaining much more attention. Now, what is this checkpoint mechanism? This checkpoint mechanism to restrict our body's unwanted immune activations. Now, tumor exploit this mechanism to evade the immune attack. We have in our body, our defense system has a very specialized cells that is we called the cytotoxic CD8 T lymphocytes. And these cells has the ability to specifically target the cancer cells to kill them. Now, when these T cells reaches the tumor site to kill the tumor cells, tumor express high amount of these immune checkpoint molecules like PDL. And this PDL then bind with PD-1 receptors present on the T cell surface. And via these interactions, tumor causes T cell functional paralyzations and promote T cell exhaustions so that the T cells cannot kill the tumor cells anymore. Now, these PD-1 therapy inhibitors or immune checkpoint inhibitors, they prevent that negative binding between the tumor cells and the T cells and rescue the T cell functions. FDA in recent years approved many immune checkpoint inhibitors for different forms of cancer, to treat the different forms of cancer. And in India also, DCGI approved the use of Keytruda, a PD-1 inhibitor, for the treatment of different advanced forms of cancer. However, for this discovery of immune checkpoint mechanisms, immune checkpoint inhibitors, Professor Allison and Honzo, they received Nobel Prize in 2018. However, this immune checkpoint inhibitor therapy also causes lot of immune-related adverse events. Although most of the events are quite manageable, but some are very severe, like type 1 diabetes or some neurotoxicity. This therapy also is very expensive, especially for low and medium income countries. And most importantly, the response is very heterogeneous. For hematological malignancies, the response rate is quite good, but not for solid tumor malignancies, which is only 20 to 30%. In our laboratory, we searched the reasons behind this partial response and very heterogeneous response. And we found that there are two types of exhausted T cells present inside the tumor microenvironment. One is PD-1 positive, TCF1 positive, progenitor exhausted cells, and another one is PD-1 positive, TCF1 negative, terminally exhausted T cells. And these terminally exhausted T cells, they can generate very aggressive cancer stem cells. Now, these cancer stem cells, they are very minor populations inside the tumor, but they are very powerful. They can create all these therapy failure, metastasis, and relapse. They are the major reasons of this metastasis and cancer relapse. And most of the conventional therapy fail to target these cancer stem cells. We studied advanced breast and ovarian cancer patients and found that these cancer stem cells and these terminally exhausted T cells are present in very high amount in advanced form of cancer in comparison to less advanced form, and the cells isolated from the adjacent tumor sites. And moreover, these cells are very high amount in metastatic cancer patients in comparison to non-metastatic cancer patients. And here we incubate the progenitor and terminally exhausted T cells with the tumor cells and in presence of PD-1 inhibitor. And we found that the PD-1 inhibitor can rescue the functions of progenitor T cells, and there is a decrease in stem cell proportion. but they fail to rescue the functions of terminally exhausted T cells, and there is an increase in cancer stem cell population. And when we inoculated these cancer stem cells influenced by terminally exhausted T cells in nude mice to check the tumor growth, we found the higher amount of tumor growth. And most importantly, the CSC causes high degree of liver metastasis in all the mice. So we recently published this work that this PD-1 therapy resistant terminally exhausted T cells can generate very aggressive cancer stem cells that can causes liver metastasis. So this scenario, this work demand a non-toxic CSC targeting low cost anti-cancer therapeutics. So here we tested our neem leaf glycoprotein. We prepared this glycoprotein from dried neem leaf powder, and biochemical tests suggest that it is a glycoprotein. It binds with the carne column. 33% is carbohydrate and rest are protein, and both this carbohydrate and proteins are required for its optimal functional activity. And recently we published that it binds with the dectin-1 glycoprotein receptors for its effect in cellular level. And in SDS page, it gives three bands, and in HPLC, it gives a single prominent band. And our previous report suggests that it can significantly restrict the sarcoma, melanoma, and carcinoma tumor growth in murine model, as well as it enhances the survivability of tumor-bearing host by mainly regulating or modulating the dysregulated or altered immune response in tumor-bearing host. It mainly activates the cytotoxic T cells. It also modulates the other immune cells as well. Next, we checked the effect of NLGP on CSC, and we found that after tumor establishment, we treated mice with 3 doses of NLGP once per a week. And after that, we isolated the tumor, and you can see here that in NLGP treated group, the tumor growth is significantly restricted, and it also restricts the accumulation of terminally exhausted T cells as well as cancer stem cells significantly. But next we studied how it reduces the frequency of cancer stem cells. So we perform an in vitro co-culture assay, but we found that NLGP only modestly decreased the CSC frequency. That means NLGP has no direct killing effect towards the CSC. It only modulates the T cells effect, but it can modulate the functional attributes of CSC. In case of tumor sphere formation assay or soft agar assay, you can see that in NLGP added group, you can see less number of tumor sphere and less number of colonies. And not only that, it also downregulates the expression of PDL1, which is a negative molecule expressed by the CSC. The green histogram represents by NLGP, and it also downregulates the expressions of NRP1 and PGP, which are associated with multi-drug resistance. These are the efflux protein. And NLGP significantly downregulates expressions of both these molecules. And it also downregulates the very important transcription factors, CSC-associated transcription factors, OCT4 and SOX2, and prevent their nuclear translocation. NLGP also rescue the T cell functions. It downregulates all the negative molecules like PD-1, CTLA4, LAG3, TIM3, and it upregulates some molecules that are required for proper T cell functions like IL-2, IFN gamma, LAMP1, and CD62L. Now, the question is how NLGP educate these cytotoxic T cells. And we find that in case of T cells, when T cells are in tumor microenvironment, they express high amount of dectin-1. And NLGP bind with the dectin-1. And here we tagged NLGP with green fluorescence dye FITC. And you can see here the nice binding of NLGP with T cells. But when we block or downregulate these dectin-1 expressions using dectin-1 specific siRNA, the binding of NLGP with T cells was completely prevented. But that not happened with the control siRNA. And next, we validate the binding of NLGP with dectin-1 is really responsible for, really required. So here you can see that where we add the dectin-1 specific siRNA, the NLGP mediated decrease in CSC populations get abolished. Again, there is an increase in CSC populations, and there is again increase in all these CSC associated molecules like PDL, OCT4, MRP1, and PGP. So next we studied the downstream signaling after binding of NLGP with dectin-1. And here we find that NLGP actually upregulate Notch1 ligand expressions on T cells like JAG1 and DLL4 and upregulate Notch1 expressions in CSC. And this Notch1, DLL4 and JAG1 binding actually causes the phosphorylations of a protein called RB, which is a very important cell cycle regulator protein, which pushes the cell from G0 to G1 phase. Now, importantly, CSC reside in the quiescent G0 G1 phase. And that is why the conventional therapy failed to target the CSC, because conventional therapy supposed to target the proliferating cells, but CSC reside in the G0 phase, G0 G1 phase. NLGP, by modulating the Notch1 signaling, it pushes the CSC from G0 to ACE phase. And you can see here, there is a significant upregulations of ACE phase, like from 3 to 18%, and there's a downregulations in G0, G1 phase from 72 to 50% with NLGP treatment. And accordingly, we found these upregulations of a very important molecule, cyclin D, which is again a cell cycle regulator protein, and as well as phospho-RB, which is specifically associated with G0, G1 to S phase transition. So next we add S phase targeting molecule along with NLGP. And here you can see the S-phase targeting molecule drug like 5-fluorouracil we used here along with NLGP significantly restrict the amount of CSC. CSC cannot kill the, not CSC, sorry, NLGP cannot directly kill the CSC. But it pushes the CSC from G01 to S phase, and then the S phase targeting drug 5-fluorouracil can kill the CSC, proliferating CSC. So the combinations of NLGP and 5-fluorouracils reduced significantly the amount of CSC. And this results reflected in NLGP-mediated restricted lung metastasis. You can see here that with B16, F10 melanoma and Lewis lung carcinoma, the nodule formations in the lung are significantly prevented with the NLGP treatment.
Dr. Anamika, you have 2 more minutes more.
Yes, sir, I'll finish. So next we validate these observations in mouse tongue carcinogenesis model, which is resemble the human carcinogenesis in different stages. So here we painted the mouse tongue with 4NQO and up to 300 days in every third days, and then we treated the mice with NLGP initially for 4 weeks, and then on day 100, day 200, day 250, and 300 days. And we found that with 4NQ treatment, we see the dysplasia, hyperplastic changes, and finally most of the mice develop tumor at the later phase. But in case of NLGP, this tumor growth was significantly prohibited in all the phases. And you can see here that with the histological staining, this is the normal control tongue. Here the tongue, the architecture is, and in case of 4NQ, there is a thickening of tongue layer and the formation of keratosis or honeycomb structures. In the later phase, this structure detached from the basement layer and migrate towards the muscular layer. But in case of NLGP, you can see the normal tongue architecture maintained very nicely. In the later phase, we see some kind of distortion, but significantly reduced. And as well as we find the NLGP treatment prevent the stem cell niche formations. Here we stain the tongue sections with CSC markers, and you can clearly see that it is very much visible that the CSC niche formations get prevented with the NLGP treatment. So in conclusions, we can say that NLGP is very much effective in rescuing PD-1 therapy resistant CD8 T cell functions. It is effective in reducing the cancer stem cell virulence. It exhibits the therapeutic efficacy along with S-phase targeting drug and is very much effective in reducing the tongue carcinogenesis. Now, what next? To bring the benefit of NLGP from laboratory to clinics, we have to cross several hurdles. So we have tried, the biochemical characterizations are partially completed. We identified the gateway of NLGP. Now we propose the testing of safety efficacy in GLP setup and NLGP formulations for clinical applications. Now still we need permission from different regulatory bodies or ethical committee and permission from DCGI or CDSCO. After that, we hope that we will reach the clinical trial after crossing these hurdles. But for that, we need help from pharmaceutical industry or biotech industry. And they are the contributors. Mohana actually did most of the work with CSC, and Saurabh Juhina, they performed the work with tongue carcinogenesis model. Nilanjan studied the NLGP and lectin-1 interactions, and they are the others who are actively engaged in our group with the Neem research. And they are our collaborators. And I wish to acknowledge Director Naipar Mohali, Director CNCI Kolkata Animal Facility, and different funding agencies like DST, DBT, ICMR, DHR, CSIR, UGC, and all my lab members, all our team members, and finally the WNO organizers. And thank you.
Questions we will have later on. Next is Dr. Gopinath.
Dr. Gopinath is an assistant professor, Department of Chemistry at SRM Institute of Science and Technology, Chennai. He holds a PhD in chemistry from Indian Institute of Technology, Madras, and he has authored various articles and is guiding MSc as well as PhD students. Now over to Dr. Gopinath.
Thank you, Balaji sir, for the kind introduction. But I could not see I could see my slide on the laptop. Very good morning to one and all. I take this opportunity to thank Vikas sir, Nirmala ma'am, and Nagini ma'am for giving me this opportunity to stand before you. So basically, I'm a chemist. So looking at molecular aspects of Neem natural products. So before I get into my PPTs or the real work, I really wanted to narrate why do we use semi-synthetic approach basically. So that's what my title also says, that synthesis of semi-synthetic. So semi-synthesis is more powerful in my opinion because so semi-synthesis, half synthesis, like what chemists do in the laboratory, half comes from the natural resources. So why is it so powerful in my opinion? Because in chemistry, creating complex architecture take ages, but Mother Nature does it, very easily. But the disadvantages in natural product is we will not be able to have the diverse skeletons, or we will not be able to engineer those molecules through biosynthetic pathways. So I take the opportunity of complex architecture from Mother Nature. I use chemistry as our tool to engineer the complex scaffolds. So that is the whole approach what we are doing it. So this we have learn even at a protein level. So expression of proteins can be done if all the protocols are fine. The proteins can be expressed in a week's time, where chemical synthesis will take 1 to 2 years. But engineering the protein through enzymatic process is big a challenge. So at the bottom line, I wanted to say that semi-synthesis is a very powerful tool combining natural products and chemical tools to modulate our molecules of our interest. So this is my lab portfolio. So we are working on various natural products, as you can see from the slide. Starting from ginger, medicinal mushrooms, but as you can see that most of the compounds are neem related. So I will be talking about 4 to 5 selected molecules here: nimbin, nimboloid, azadirone, epoxyazadione, and azadiradine. So I'll just give it a glimpse of how much we are able to obtain from the waste. So for example, when I start with 1 kg of neem seed kernel, roughly I will be able to get 1.1 grams. As most of the expert knows, this number always varies, but this is an average quantity in which we are able to isolate. So quickly very briefly, I will talk about the isolation process because this is very robust. And also many people have done this. So we take 2.5 kgs of neem seeds, and then you remove the kernels. So once you do the extraction, using various solvents, we use methanol as a primary solvent. We end up with 60 grams of crude, which we will take up for further purification through typical column chromatography. We end up with nimbin. So it's not like one shot purification will be enough. We will have multiple purifications. Sometimes we also use crystallization techniques to get the pure material. So this is how nimbin is got from the neem seed kernel, and then nimboloid we have obtained from neem leaf. So since I'm a chemist, I would like to highlight the fascinating correlation between the structures within the natural products. If you look at nimbin and nimboloid, by and large 90% of the skeleton is the same, except that the red ester. So we have 3 functional groups: 1 ester attached with primary carbon, 1 ester attached with the tertiary carbon, and 1 we have an acetate group. Okay. So as you can see, there will be a lactone formation from the nimbin. So you just do a cyclization, you'll be able to get a 5-membered lactone that is nothing but nimbin to nimboloid. I oversimplified it, but in nature it does in completely different way. But I wanted to bring the benchmark the structure, and wanted to appreciate how we are using our chemicals to fine-tune the chemistry. So the biggest challenge with respect to late-stage functionalization we call, because the molecules are already built. So tuning the chemical functional groups wherever you want is going to be challenge. So we have 2 ester groups and 1 acetate group, and we asked our question whether we will be able to do chemistry specifically at one site at a given point of time. That is a simple question we have asked. So with that question in mind, we have initiated several optimization studies. I don't want to go into all the details. I will just wanted to give the end results only. So when we treated the nimbin under basic conditions, sodium carbonate, so what we have found is we were very selectively able to deacetyl the nimbin. So this is called deacetyl nimbin, which is again a natural product which is already in the neem. But if you take the neem, the percentage of neem and the deacetyl neem varies based on the seed, the age of the seed in which you are going to pick it up. So if you give me neem and ask me to make the deacetyl neem, I will be able to do that. If I want, for example, I'm getting more of deacetyl neem in nature, if I wanted to reverse back, that also can be done. So we have obtained compound 2 in almost quantitative yield by simply stirring with sodium carbonate in methanol. So since it's a secondary hydroxyl group, so any chemist would love to do an oxidation on it, because installing carbonyl compound at a complex molecule will open new avenues for further chemical manipulations. So what we have done is We have tried various oxidation, oxidizing agents, all work well, but DMP is nothing but desmartin periodinate. So you oxidize under DMP, we end up with a ketone. So this particular process, we have patented it. It got published, but we are waiting for the grant to come. So the same Neem, when we are treated with potassium carbonate, what was happened is we could get molecule 2 initially, but prolonged reaction conditions, we also obtain compound 4. So compound 4 is nothing but deacetylated and also hydrolyzed specifically at a ester which is attached with the primary carbon. So in this case, we have done later a lot of optimization. And if you are stirring Neem with potassium carbonate over 24 hours, we get exclusively compound 4. So the beauty of compound 4 is we were able to deacetylate, and very specifically we are able to hydrolyze only the one ester of it. But how do we confirm this? It is very difficult to confirm by NMR because the other possibility is the
माटी पप्पा
hydrolysis
बहुत
of
इतनी चिंता नहीं
tertiary
करवानी फटाफट जवाब देवानु
in the
थोड़ी घड़ी
first
माटी तो
ring.
जवाब देवानी
So,
आपने
we have done a lot, and then we also finally obtained the single crystal XRD that is shown as a crystal structure of 4. So anamically characterized it, and that also when we exposed to the oxidizing conditions, we have obtained compound 5. So if you look at compound 2, 3, 4, and 5, we have managed to fine tune the functional groups within the Neem scaffold. So if I wanted to hydrolyze the ester,
जो हटे ना थोड़ा
the tertiary ester in the very first ring, it is not possible through Neem route. So what we have done is we have taken nimboloid. As a chemist, we know lactone is very labile. You hydrolyze the lactone in the presence of potassium carbonate. What we end up with is acid at the first ring. The ester at the second ring remains undisturbed. Even acetate group remains, undisturbed if we control the condition, but we can also deacetylate. So basically, simple base-mediated transformation and oxidizing agents, we will be able to play around very site-specifically all the chemical transformations.
नौ
Complex skeleton. Okay. So we also did an oxidation on this one. So the bottom line is the chemistry is pretty simple. There is no very high-end reagents involved in it, and the scale are also working very good. And we are also able to engineer very, very specifically the functional group across Neem. So quick NMR. So I wanted to make it simple. Let me try it. Okay. So basically I have an acetate group here, which appears at 2.2 ppm. I have an ester over here, and then I have ester over here. These three single peaks in the first spectrum represents those. So this is acetate, this is ester of one, ester of second molecule. So when we have done only deacetylation, this particular singlet peak is missing. I want to oversimplify it, but these are the analytical data I wanted to just give in a very robust way. We have done all the characterizations. So this was only one deacetyl, and then after we treated with potassium carbonate, one of the esters got hydrolyzed. One ester is still intact. So we have obtained the single crystal XRD for this, but we have done extensive solution phase NMR, etc., which is typical characterization done by an organic chemist. So what next? Because the skeleton have a lot of functional groups. For example, this alpha beta unsaturated group is pretty versatile, and one can develop quite a lot of chemistry with that. But the problem with this particular molecule, this functional group is as soon as I touch this beta carbon, create a chiral center, so that will be a mixture of compounds. Play safe because of already we have 4 rings along with 8 to 9 chiral centers. We will not be able to. So what I've proposed is a Lucci reduction, which is known for its selective reduction. So when I do only sodium borohydride, I end up with a mixture of compounds stereomerically. So when I use cerium chloride, which is an established chemistry by the Lucci, it's a naming reaction, very robust reaction. So what I would expect is an allyl alcohol, but I have put it in the red because again you create a chiral center over there. It's not very easy to control chiral center, but we already have the scaffold, which probably will help to get a site selective reduction because Lucci is, we have a model mechanism, the cerium chloride will complex, and there is a lot of mechanism. One can expect some selectivity.
Dr. Gopinath, you have 3 more minutes, please pass.
Okay. So what we have done is this reaction, and then what we have found is the cyclization over here, which is driven by the the overall skeleton of nimbin, and then this particular lactone got reduced in situ, and then we also obtained deacetylation. So all these 4 reactions happen within 30 minutes. So it's very specific, very, very clean reactions. And no one will really believe if I say like all these 4 reactions happens in one pot with various analytical techniques, unless I capture through in single crystal XRD. So we have struggled 2 years to get this single crystal XRD. So we have predicted through NMR, but NMR can lead to so many, speculations. So this is a single crystal XRD of the very new material which we have obtained. We are in the process of filing a patent across this. So can we use the same, strategy for the nimboloid? The simple answer is no, because nimboloid, there is another 5-membered ring. The geometry is not going to allow these particular cyclizations. But this first step, this Lucci reduction, until then, if I treat nimboloid, it will stop in this stage only. Okay. So nimboloid, we have tried different chemistries, that is Bailey-Silman chemistry. We wanted to do introduce a double Michael acceptor in addition to the lactone, which is important for its various biological activity. We have done, and we have also screened. I'm not going into much of the biological details. So we have also employed Rayleigh oxidation. So since in the we have a few minutes, I wanted to only tell you that this Rayleigh oxidation can be employed for allylic oxidation, also on the oxidation of 1,2-dicarbonyls. So using this chemistry, we have employed a variety of natural products within the Neem, and we were able to very, very selectively oxidize allylic position of epoxyazidadione, and this chemistry works beautifully well with other skeletons as well. And one particular product we have crystallized and shown the structures unambiguously. So what next in this particular thing we are looking at? We have made a variety of Neem natural products. We also screen against various antimicrobial strains. And for our surprise, only one specific semi-synthetic molecule is showing some degree of, like, it's a good activity against Staphylococcus aureus. That also tells the power of semi-synthetic approach. So we also proposed NOS inhibitors along with this. So we wanted to go one step. So this particular grant is technically approved by DBT. So we will be looking at diabetic nephropathy of these semi-synthetic molecules using in vivo models. So with this, I thank you one and all for your kind attention.
So next we will have Dr. Devendra Kumar.
Dr. Devendra Kumar is the head of R&D Center at PJ Margo, Bangalore. He has more than 3 decades of experience in Neem industry for process and formulation development. He has developed more than 40 biopesticides, biostimulants, and biochemical agents which are in the public domain, marketed in various countries. Dr. Devendra Kumar.
Good morning, one and all. I would like to thank first World Neem Organization and especially Dr. Balaji and Dr. B.N. Vas for giving me opportunity to make a presentation here. I will discuss with you about the Neem, like what are the work has been done in last 155 years on Neem. What is the importance of Neem? And how Neem has many uses. How we can improve its effectiveness by different ways. In last 155 years, more than 19,500 papers have been published on Neem. Most of the work has been done during 2021 to 2020. Each part of Neem has been explored. Most of the work has been done on oil, seed, fruits, and roots. Various biological properties of neem has been explored, such as antimicrobial activity, antibacterial, pesticidal, and so on. There are 4,344 patents on neem, and maximum number of patents have been filed between 2021 to 2020. And I am happy to tell you that maximum number of patents have been filed in India, followed by China, and then PCT and US patents. There are various categories of neem where most of the work has been done on pharmaceuticals, then pesticides, personal care, fertilizer, soil amendment, and other things. More than 6 to 9 crore trees are there in all across the world, and highest number of seeds are found in India, around 2 crore. In that also in UP, highest number of seeds are found. India has a scientific gold mine. We can use neem in medicine, pesticide, and in so many ways. For agriculture use, azadirachtin A and B have been identified as a most potent active ingredients. There are more than 3,000 compounds identified in neem. First report on neem was reported against locust in 1959, and first report on antioxidant activity of azadirachtin was given in 1962. Now, it has been found active against 413 insect species. Neem does not develop resistance against the insects. Why it does not develop resistance? In chemical pesticide, we have only one chemical or two chemicals. The insect can easily develop resistance. Whereas in neem, there are hundreds of compounds. It cannot develop resistance with so many compounds. And the beauty of neem is it is very safe to farmer-friendly insects. Why it is safe to farmer-friendly insect? When it is killing all the other insects, why it is not killing honeybee or other farmer-friendly insects? Because honeybee will not eat the leaf. It will take the nectar and run away. Other insects will feed on other insects. They will not feed on the leaf. The mode of action of neem is well known. There are so many publications on mode of action. But how we can improve efficacy of neem-based products to get most benefits out of this? We did some experiments on synergistic effect of azadirachtin with other products, and there is a lot of published information also on synergistic effect of neem in pharmaceutical products, personal care products, and agriculture products. We can make use of this information. There are 626 publications on synergistic combinations of neem. For pest and disease control, people have used neem oil, orange oil, neem oil with pyrethrum, neem oil with the mint, neem oil and marigold and coconut oil. So many combinations are available. For nematode control, people have used neem plus artemisia, neem plus chili plus cinnamon oil. For antifungal activity, neem plus castor oil, neem plus sesame oil, and so many combinations are there. Most of the work has been done in pharmaceutical. There are so many uses in pharmaceutical, like neem plus fenugreek plus turmeric, neem plus eucalyptus oil, neem plus coriander, neem plus paraffin, neem plus mentha. There are so many information on combination of neem with other plant extracts. In personal care also, neem plus coconut oil plus vitamin E, neem plus aloe vera, neem plus karanja. There are so many information. In vet care also, so many people have used many combinations like neem plus water nagunda, neem plus fenugreek, neem plus karanja, and so on. As a disinfectant also, neem plus lemon peel, neem plus citric acid and curcuma, and you already know neem has been used a lot in urea, neem cake plus azadirachtin. Neem also finds very good use when we use it in combination with microbial pesticides like trichoderma harzianum, neem plus Beauveria baicana, neem plus Bacillus subtilis, neem plus Bacillus amyloliquefacens, neem plus BTK. We also did our trials with neem plus BTK. We use Eco Neem Plus, which is manufactured by PJ Margo. which PJ Margo is the first company in the world to produce azadirachtin technical. We use this Econim Plus from PJ Margo along with Delphin, which is BTK product, and these are our results. First one is control. This is only water treated leaf. Second one Econim Plus, then Delphin plus Econim Plus. So we have put 3rd star larva of Spodoptera, and you can see how it is eating the leaves. In control, it has control if finish the leaf. In Econim Plus, you can see control in Delphin control and more control in combination product Delphin plus Econim Plus. You can see without treatment, completely finished. There is 71% control in Econim, 84% in Delphin, and 93.2% in Delphin plus Econim plus combination. We tried different combinations for dose dependency test and LC50, LC30, and LC20 values. Econim Plus from starting from 1.8 ppm to 30 ppm, and Delphin from 125 ppm to 2000 ppm. You can see Econim Plus is having control at all the doses, starting from lowest dose 0.18 to highest dose 30 ppm. This is larva plus pupal mortality. In Delphin, at lower doses, it is not working, but at recommended dosage and half of the recommended dosage, it is giving full control. The LC50 values was at 8.05 ppm for Econim Plus and 733 ppm for Delphin. Then we tried so many combinations based on LC50 value plus LC50 value of Delphin plus LC50 value of Econim Plus, then LC30 plus LC20 and different combinations. You can see Econim Plus at 3.73 ppm and Delphin at 365 ppm. You can see individually they are showing only 20 and 30% mortality. When we combine together, activity goes up to 70%. Similarly, in next graph, you can see Econim Plus mortality 30% and Delphin mortality 20%. When we mix together, the combination has given 83% mortality. When we have used as LC50 values of Delphin and Econim Plus, both are showing 50% mortality, but when we mix together, activity goes to 100%. That reduced dose we can use, we can get 100% mortality when we use in combination. This is growth inhibition. You can see Econim Plus is showing growth inhibition because it has multiple mode of action. Insect will not feed and it will slowly it will die. Whereas Delphin is not showing growth inhibition. It does not show growth inhibition. It directly kills the insect. So how it kills? Delphin will go inside the midgut of the insect and it will form cryotoxins. And with these toxins, insect will die. We also checked biological activity, like compatibility of tank mix in CFU count in the tank mix solution, and we found there is no degradation of CFU count if you keep up to 12 hours. Chemical compatibility, we checked, we kept this solution for 12 hours and checked azadirachtin content. You can see up to 5 to 7 hours, there is no significant difference, but it suddenly comes down. It means we have to use it as fast as possible once we make a mixture. So both azadirachtin and Delphin were found active against Spodoptera, and combined effect of Econim Plus and Delphin gave 90 to 100% mortality of third instar larva. And extended studies on compatibility of these two biopesticides indicated that combination is stable up to 7 hours after tank mixing. This can also be used as an IPM tool. Thank you.
Thank you, sir.
So the next presentation is from Dr. Kumaran, please.
Dr. Kumaran is a professor, forestry at Tamil Nadu Agricultural University, Tiruchirappalli. He has been working with Neem for more than 30 years. His PhD thesis was also on Neem. He has published various articles and he has been guiding MSc and PhD students on various disciplines. Over to Dr. Kumaran, please.
Check. Hello.
Please come. He will come.
Yes, done it.
I'm getting to.
Good morning, everyone. Thanks for Dr. Balaji for introducing me, and I'm highly thankful to the WNO for giving me an opportunity to share my experience of 30 plus years in Neem research, particularly on the genetic improvement. And I started my research in Neem in 1989 from my MSc. I continued till now, and then we got opportunity to work with industries. Any research without industrial collaboration is not going to happen, but we have done it successfully. And almost I attended all 8 Neem conferences since 1992 when Bangalore they started the Neem conference. That was my first presentation on Neem. And then my area of thing is that mainly genetic improvement and productivity in terms of Neem seeds, which is a very important thing to be discussed. And not only that, productivity with high oil and high azac. This is the thing we have done. The entire research on my career, and then what we have done for the since last WNO at Bangalore, what we have done. In Bangalore, I shared what we are going to do. In the last 5 years, we achieved, we are going to share here. I'm not going to the details of the Neem basically. The one thing very important that Neem is, there are few trees which can be used, each and every part of the tree can be used. One such tree is Neem. It has multivarious utilities, but it's not rightly taken to the farmers or supported them how to make each and every part of the product to be useful. I have classical examples of few people who used to contact me, and one such small youngster from Tirupur, he used to export 1.5 lakh Neem sticks to US regularly. So there are a lot of opportunities for Neem to get involved. If you see the statistics of the global Neem extract business, it is continuously increasing. It's never come down. Equally, the price of Neem seeds also increasing. But why this price is increasing? Because two important things that the number of naturally grown Neem trees are very, very high, where we fail to have a process of proper collection and processing. Even though we have huge amount of, I'll share in the subsequent slides. See, CAGR, the cumulative growth rate of 15.09 is very rare to find in biological things. I am working parallelly in other trees and other compounds, but Neem is having an excellent projection for the years to come because there's a huge demand not only in the agriculture part, but also on the therapeutical and pharmaceutical sides. So when we started things in 2010, we made a small assemblage. When I started my Neem research in 1989, it was 34 million Neem trees in India. It has come down to 31 million, and present it is 25 million. Means many trees are being felled for developmental purposes. But if you see the other scenario, the total Neem fruit collection from these 25 million Neem trees is only 3 lakh tons. And then conversion of these 3 lakh fruits into oil is very, it's not even 10%. That means the quality of the seeds collected from the ground and processing, we lose almost we are projecting 40% oil content, but oil content recovery is very less. I was working with GNFC where the recovery of oil content is less than 10% because the quality of the fruits, seeds they are collecting and supplying to the industry is very, very less. I'll come to that also. We'll intervene there and we supported them. And this is the production potential of a normally grown neem tree. Normally, it is occupied its huge amount of green biomass. I'll come to that point, which is very, very important point. And then which normally have 50% leaves and then less trees, wood and fruits, which are converted into things. But the actual Indian scenario, we have 450,000 tons of seeds are only collected. Collection is only even less than 2%. Most of things are going as waste. And from this 450,000 tons, we are getting only oil 90,000 tons. Yesterday, the production for urea coating is very high. But even then, this oil is not only for going for urea, but neem oil is being used for many other purposes. That's why we are having a shortage of supply to the urea coating. Normally, 10 years old neem trees, which are being mostly grown as isolated or self-grown trees, it is only 50 to 60 kg fruits. When on conversion, we'll get on to only 6 to 7 liters of oil per tree. Coming to the oil content, we are say that's what I wanted to highlight here. 38 to 52% of the oil content on the lab scale. But when you go to the industry, where the industries are collecting or buying from the mandis, the recovery is very, very less. And then aza content, if you see 0.01 to 0.9, David Morgan's paper. But my work in my MA PhD work, we found that there's a scope that we have found from one tree from Rajasthan, we got 1.4%, for which I got a national award from Jawaharlal Nehru Award from the ICAR. And then we started thinking that the very less amount of oil content, aza content reported was because of the lack of sampling. So any sampling has to be done in wide area. Not even still now, to my knowledge, we are not sampled even less 2% of the total neem trees across the country. But I will share my recent reports. There is every possibility that we may reach 2 to 2.5% in the years to come. And this is the phenology of neem tree. Most of you know that. And one important thing is that yesterday also there was a huge discussion that aza is genetically controlled or not, or climatically. Aza is 100% genetically controlled. And the temperature or rainfall normally deters or influences the flowering and fruiting of neem. Either it advances or delays. The productivity is directly correlated to the silviculture. The very important point here is the reproductive biology. The natural seed set in neem is only less than 1%. We get more than 150,000 or 2 lakh flowers per tree, but conversion is only 1% of the flowers are converted into. And because the flower shedding or flower conversion is directly correlated to the rainfall, temperature, humidity happens the last 4 months before flowering, or the last year's rainfall also. The climate is directly correlated to the flower to fruit set ratio. And then the main pollinator is honeybee, and flowering, everything is there. And the maximum oil and aza depends on the stage of fruit collection. That is very, very important point. We had a breakthrough, and it is proven also. And pollination, unless few literature says it's self-pollinated, it's 100%, 95% cross-pollinated, and 5% is self-pollinated. And even in the 5%, it is because of the incompatibility of pollen that is less form. And then we also try to understand why the fruit set is very less, because fertilization happens in all the most pollination happens, but during fertilization, almost 60 to 70% abortion happens. After fertilization, the seed gets aborted, ill-filled, or even premature fall will be there. So normally, even we are able to increase the flower to fruit set ratio to 5%, the productivity is going to be almost 100% or 200%. And neem, every part is used. Major thing is we are using seeds, oil, azadirachtin. And the recent past, when you go for plantations, gum is having a beautiful market, excellent market. And we also can rear honeybees in the neem plantations, which is an additional income to the farmers. I'll touch that point. And finally, after 30 or 35 years, if you want to fell the tree, we have excellent wood for the market. I'm not going to the chemistry much, but one thing is azadirachtin, the novel compound. I had an opportunity to work with Dr. Smatter during my PhD because when I found that there was 1.4%, 14,000 ppm of azadirachtin, my chairman didn't allow me to publish it. And then we sent the sample to Dr. Smatter's lab to confirm twice, and then we published a paper. That was a breakthrough for us to continue in the azadirachtin. And this is the variability reported across thing. And the variability is mainly between trees. That is, of course, yesterday also, I think the co-chairman, when Rajarajan was presenting his paper, 150 gene is contributing directly to the formation of oil and azadirachtin. So it is genetically controlled. But a huge lot of papers have been published in azadirachtin. But it is now very clear that we can try to identify a plus trees with high aza and then promote through progenies, we can get the results. And the issues why Neem is not able to, because neither it is neither wide nor properly cut. We cannot see Neem trees in the forest ecosystem. It is mostly scattered trees on farmlands, avenue trees, and mostly self-grown also through bird droppings. And then very important thing is that unorganized seed procurement collection. We worked with the GNFC to intervene them. We trained their self-help groups to collect the fruits directly from the ground at the green stage, and then process, and then seeds were got. The recovery from 7% of oil increased to 23%. And GNFC also happy, and the self-help groups who were being given 8 rupees per kg were given 30 rupees per kg later. So there's a direct contact, and small scientific interventions will help people to work on the Neem collections. And then a very important thing is there is no organized large-scale plantations in India except few, and then we started working with industry. The genetic improvement, what we have done so far, we collected Neem samples from laboratories across the country, so many samples, and then tried to map the genetically superior, phenotypically superior samples in the progeny evaluation trial. And then we tried to establish 2 field gene banks, one in our institute and the other in the industrial part. And then we parallelly promoted the selections to the plantations, industrial plantations directly, so that when the results comes, it will be better. And finally, with the results, encouraging results from the industries, we could be able to introduce, launch hard contract farming, which is very successful in Tamil Nadu now. And this is the what I told, the fruit collection. Fruit should be collected only from the ground, not from the ground. And even if you want to collect from the ground, there should be a stage with which you have to collect the fruits. And then the few plus trees we have done with high oil and azadirachtin, and a few I want to share. Particularly, I want to share this slide where we could able to find last year. There are 4 important trees from both Tamil Nadu and Telangana, which recorded more than 1% of azadirachtin. It is last year also we collected one sample which is 1.23. We'll go for 3 years continuous assessment of the azadirachtin. Once it is consistent, then we'll promote them to the our gene bank. And we try to concentrate on the basic success of any plantation depends on the nursery quality. So we took utmost care in the entire SOP of the nursery. We developed a nursery with all packages, and then we taken to the industrial plantations. I'll show the plantation. So the success is we are too important because genetically now we have trees with good oil and azadirachtin. Progenies are directly from the mother trees. The second part is silviculture is very important. We should take care right from the nursery, the planting, how we are going to planting, the techniques of planting, how we are going to give the inputs, and how you are going to maintain for the first 2 years. All these things, the silviculture play a very vital role in the success of the plantation in terms of its not only the seed productivity but also in terms of oil and azadirachtin. And this is the progeny evaluation trial we have done at Metropolium. It's a progeny evaluation trial. It will be converted as a seed orchard later stages. And this is a basic plantation management. Spacing depends on what you want. We are going for high density planting. Normal recommended plantation packages normally we go for 4 by 4 meters, 250 trees per acre. But we are also trying a combination of 4 by 2 meters. It is under trial, because when you reduce the crown size, the total biomass, the energy required for the total biomass for a bigger crown will converge to a smaller crown. The energy will be converted into fruits. We are aiming for even 4 kgs of seeds per tree. It is going to be a very successful productivity, but I think we will go up to 10 kgs per tree. And very important thing, as I told earlier, it was a self-incompatible. That's an issue in the pollination. We started the supplemental pollination in the plantations. It is encouraging. Last 2 years we have started. It's a 3rd year. Probably we'll get that. And natural fruit set is only 1.1 to 2%, where we get 0.4 to 0.7 tons in the natural seed set. Whereas when you go for spray pollination, the fruit set may go up to 8%. It's a first preliminary trial. We will be confirming that. And the yield will go up to 2.5 tons per acre. It is very, very, very high. And maybe this is one of the best crop farmers can think about because the seed price of neem seed price is almost 120 to 180 last year. So even getting 1 ton per acre after reducing all the inputs, probably 50 to 60,000 per acre a farmer can easily get. The plantation we raised with the industry, one of the industry we are collaborating. It's one of the biggest plantations. Yesterday there was a discussion. There is no plantations in India. Tamil Nadu, we have beautiful plantations over an acre of 800 plus acres, along with the industries. It is well done with followed all the SOPs and properly managed, and we got the results. And the flowering was in the 3rd year, and we clipped the 5th thing, and we allowed the flowering in the 4th year and 5th year. This is the 5th year, probably will be getting. Normally in the neem, general neem or the self-grown neems normally comes to flower in 7th year, but now we are reducing 4 years to reduce that. It is because of the parental selection and along with the silviculture. Both will help to increase the flowering. And not only that, the plantation, when you go for a proper management, the synchronization of flowering and fruiting is happening. That is very important factor to be considered. Otherwise, normally the flowering and fruiting takes longer time from April to September. But here the synchronization have 90% of the flowering and fruiting happens in a very short term. That is easy for harvesting also. That is one of the very important advantage. And these are the few plantations we raised in 3 areas, totally around 600 acres. And not only that, we also tried to rejuvenate an existing plantation of 12 years old plantation, which was not at all productive. Probably Dr. Balaji and myself, we visited initially to the plantation. And then we intervened. Not only that, we just cleared all the undergrowth. We tried to prune the trees, give some irrigation with some manure, and then the plantation started flowering and fruiting. So small interventions is very, very important for flowering. Particularly, neem responds very well to pruning. We have to do the pruning regularly. We have to standardize that. We have done that also. This is the total journey. The last 11 years, we started with the plus tree selection, and then totally we now assembled 55 plus trees with the ASA. We made a benchmark of minimum of 0.8% of ASA. All our plantations are having minimum 0.8 and then maximum up to 1.2. And then we tried to go for captive industrial plantations in the, along the industry. And then based on the success of the industrial plantations, we promoted the entire plantation to the farmers field. Once we felt that it is successful, then we have taken to the farmers. It was working well. And then contract farming, I'll share. And already it was launched in 2021. Advantage of benefits of contract farming when we see the entire technical support is given by the institute. Forest College is taking that lead. And then we'll supply genetically superior progenies from other plant is properly collected, properly isolated with proper labels, and then supply quality planting stock. And then technical guidance right from the layout till your market. The entire thing will be done, supported by the institute. And then mechanization, we started working on mechanization also, because when you go for larger area, it is very difficult to collect the seeds. So based on the experience of olive harvesters from Spain and Israel, we started working on that also. The industry is doing that. We have started from harvesting is done mechanized, and then after harvest, depulping at the collection stage, and then extraction of seeds. Finally, even drying and the grading is done mechanically. Probably it will be under trial, will come into action within 1 or 2 years. And then assured price for the neem seeds through the contract agreement with the industry. That is very important. Now farmers are happy that they are getting an assured price here. Here we are directly linking the farmers to the industry. There is no middleman, so both industry as well as farmers get the benefits. It's a win-win situation for both the farmers and industry. And finally, additional income through carbon trading. We are working on carbon trading also. Recently, we have finalized all these things. Forest College Metropolitan has contacted. Every 5 neem trees will get 1 carbon credit. Probably present rate, it is $8 to $12 per year. So this is an additional income for the farmers in the first initial phase of thing. And they can, only condition is that, present condition is that they have to maintain the trees at least for 10 years. They have to sign an agreement with the carbon trading agent. They have to maintain the trees minimum of 10 years. And then increased yield and economic returns. These are the major benefits of contract farming, and farmers are slowly coming up. There are many inquiries happening from different states of the country to us to invite us to started farming. And these are the few mechanization we started. And then we launched the contract farming in 2021 at a Metropolis, a big function. 4 farmers, they are going 20 acres each. We signed the agreement on that day. These are the few farmers field where the neem plantation is going on. It is almost 2 years old. Probably next year we'll understand. And even second year, we got into farmers field, we got flowering in the second year itself. But we try to clip them and then don't allow them to fruit. And probably from this year onwards, we'll get some good results. And finally, the neem plantation for sustainability. Not only it helps the economically, but it also try to help a sustainable value chain model here, because we are trying to meet all the growing demand of needs through plantations. And in India, still we have 300+ million hectares of wastelands. So we thought that the industry can talk to the government can give, come in agreement with the, like in other countries, we can also come develop these wastelands. Sure, I'll complete. And then increases the biodiversity of the locality. The very important point that when you go for plantations, the local people will get an employment opportunity. Not only that, the biodiversity we recorded even the biodiversity in these plantations. There is tremendous increase in the biodiversity, like birds visit because pollination happens, lot of nectar, everything. Not only that, in neem plantations, we also try to put some other trees which will attract more honeybees, because neem needs a lot of honeybees for pollination. We try to include few other tree species scatteredly in the boundaries or inside, so that the honeybees will be attracted. And then it is also helps a lot of the mission of achieving 33% that green cover. It is possible to achieve that. And then environmental amelioration through the carbon credits. So finally, the way forward, we try to promote the neem plantations with the governments. There are a lot of scope, and many industries can come together or come forward to support these programs. It is easier to do. We already proven model for that. We can do that. And then we can also promote neem cultivation small scale neem farmers also through as woodlots or scattered trees. Even on bunds and boundaries, we can do that. And we go for even the procurement side. We try to train the local self-help groups or people who are involving in the seed collection from the existing trees to how to collect from the ground and how to process it. That will help to give a better price and better quality of the seeds. We can do that. And then we already developed a value chain. We are going to promote it through that. And finally, it's the right time. I'm writing a draft policy on seed procurement. I'm drafting it now. Probably I'll submit to the government in the very short thing. Yeah, how to the existing 20 million trees, there should be some policy guidelines to collect and support, because the prices, everything is different in different states. We try to bring a common base. So the proposed value chain, how we are doing that, the institute will do the R&D and develop a variety, release a variety. Farmers can cultivate the promoted varieties, and the industries can support the entire R&D part, as well as they can procure the seeds from the farmers, so that it can be complete value chain. And we also working on the giving insurance and the financial support from the financial institutions. And already we are preparing a draft on the SOP of Neem, so that insurance can be given for the crop. Some country and our institute has already done insurance for 4 important species, and now we are working on Neem for the insurance. So really, it's a miraculous tree. With my 33 years of experience in Neem, still a lot more to explore. It's a beautiful species to work with. So I'm very happy to share all my work here. So thank you very much for the opportunity. And those who are interested to do any of such plantations can feel free to contact me. Thank you very much for the opportunity.
Thank you. The last presentation in the session is an online presentation by Dr. Terry Pelez from Tanzania. Is he online, please?
Good morning, everyone.
Welcome.
My
Terry
name is
Place. This is Ms. Terry Place, online from Tanzania. Terry Place is working on Neem in Tanzania and other places. Over to Terry Place now for her presentation. Thank you.
Thank you so much for inviting me. I'm not nearly qualified enough to be on this panel, as my presentation is not scientific. It's merely anecdotal, based on one very emotional year of observation. But I feel so privileged to join you all today and begin a much deeper understanding of this incredible tree that I know will last the rest of my life. For the past 20 years, my husband Wandu and I have been running an orphanage and a 12-acre farm and a primary school in Tanzania. 15 months ago, he showed me a black ulcer under his tongue. I knew instantly that it was cancer. The biopsy took several weeks to arrange, and I turned to Dr. Internet and researched for hours on end, reading dozens of medical journal articles about which natural products induce apoptosis. I found several articles showing promising research using compounds derived from Neem to reduce squamous cell. Of course, I could never find medical-grade Neem compounds in Tanzania, but our farm is home to several hundred very large Neem trees. At this point, I had heard claims of what the mwarobaini tree could do. Mwarobaini is the Swahili name for Neem. But I had rarely tried to use it for anything. I begged my husband to put the leaves under his tongue and to drink Neem water daily. He agreed, and all day long he worked and spit Neem juice out. Weeks later, the biopsy confirmed that he had stage 3 or 4 squamous cell cancer in his mouth. The doctors wanted to remove most of his tongue. He was calm, and I was terrified. While I searched for the money to get him to India for surgery, the doctors gave him 3 rounds of chemo to shrink the tumor. Meanwhile, he kept up with the Neem and cleaned up his diet. I sat beneath one of our giant Neem trees and I prayed to the tree to help us. In March, we left for Delhi, but when he underwent the PET scan, there was no cancer evident in his body. There was still a swelling under his tongue, but the PET was clear. Doctors in India still wanted to do surgery to remove his jaw, just to be safe, they said. When I talked to other doctors, nobody could believe that only 3 rounds of chemo had made this much of an impact. One of his doctors even had the original biopsy sample to be retested in a different laboratory. It was confirmed to be squamous. After much discussion, Wandu opted for radiation only and not the surgery, and used Ayurvedic medicine to reduce the side effects from the radiation. He was a paragon of strength, and after 4 weeks, the lump was gone. We returned home to Tanzania, exhausted and humble, but hopeful that his triumph would last. Of course, we cannot prove that it was Neem that helped him, and we cannot be sure that he will remain cancer-free, so I don't tell many people about this. But in this forum, I'm not afraid to make these claims. I hugged our Neem trees in gratitude for their blessing. 2 of the things that I would like to briefly speak about today are my observations from this year. Our farm is in a semi-rural area of Tanzania with great poverty. Every day I see so many children with fungus on their skin, their heads, toes, and faces. They suffer needlessly and struggle to afford useless antifungals like fluconazole when the answer is all around them. Since returning from India, I have worked with people on healing fungus and painful shingles with topical Neem with 100% success. I've also had people laugh at me and refuse to use Neem, considering it to be backward and dangerous. As for cases like my husband's, which require ingestion of Neem, I am not naive enough to claim that Neem is a panacea or without potential side effects. I often worried myself whether he was taking too much or too little. But we believed that the potential benefits outweighed the risks, and we seem to have been correct. I think you all have a very different experience in India. But from my perspective over here, I believe that Big Pharma has a vested interest in making us fear Neem's potential side effects. Meanwhile, they are removing tongues and jaws and other body parts in the name of safety. I am incredibly grateful to be here today to learn of the research you are all doing involving isolating compounds within neem and even synthetic neem. And I am so encouraged to learn that much is available in the public domain. I wish that I had been able to find more. As a non-scientist, I hope that one day more research involving the whole neem plant is done and made accessible to more people. I believe that Big Pharma's goal of profit over actual health can create dependence on the medical system and discourages medical sovereignty. In India, people know and believe what neem can do already, but here in Tanzania, it has been forgotten and more knowledge is urgently needed. I would like to be a part of teaching people in Tanzania how they can promote their own health and medical sovereignty. When I wrote my abstract for this conference, I was still in the romance period of my relationship with Neem. I was so grateful for my husband's healing that I could see only the blessings of this tree. I have been running a charity for 20 years here, and I'm quite tired of asking people for help. I had simplistic dreams that Neem would change my life. I will be a Neem businesswoman, not a woman who begs for money. Much like my nemesis, Big Pharma, my mind was spinning with all of the options for profit: cosmetics, birth control, STD prevention, chicken feed, fungicide. I was ready to sell. Then I had a rude awakening from my dream. I learned that Neem is actually invasive and destructive in Tanzania. It steals nutrients from other plants, and it's nearly impossible to kill. How could my blessing also be a curse? Very quickly, my romance with Neem ended, and I found myself in a marriage. Now the real work had begun. My husband and I walked our 12 acres carefully this time, seeing with new eyes the destruction, the overgrowth. Although we had been careful to plant our trees away from buildings, it was clear that Neem was in competition with our citrus and dominating. How did I ever think that more was better? This all happened, by the way, only a few weeks ago, so that is why it has changed my presentation. Again, these days, I sit under my neem tree and pray. I ask it how we can work together to protect people and to create an income for our family and community, to maximize its benefit and minimize the burden. Today, I have put profit aside again while we are working on three things: management, education, and reforestation. In terms of management, we are making a survey of our 12 acres, writing down all of the location of all of the trees and the size, and trying to strategize where we can have neem and what is a safe number. We are in an aggressive campaign to dig up all of the small trees before they get too deep and hard to kill. Once we know how many trees we can safely keep, I plan to return to India to make a more serious study of neem with my daughter and get back to work developing useful and sellable products for here in Tanzania. In terms of education and reforestation, our neighbors know nothing at all of the benefits or the threats of neem to our area. So we have ordered a lot of native trees, the ones that native birds enjoy, and we will plant these and give them away and use our primary school to teach our neighbors how vital the native trees are. We have also begun to educate our school children and their parents on the blessing and curse that neem is to Tanzania. And in closing, I just wish to say that I am immensely grateful that because of Neem, I was privileged to meet Dr. Kotari and be invited to share my story and opinions with all of you. Thank you. That is all.
Thank you. Thank you for this. So now the session is open for the discussions and any queries. You can introduce yourself and ask the question from any speakers, please. Yes, please.
Hi, I'm Sharad Singhania from PJ Margo. My question is to Dr. Kumaran. Excellent presentation, sir. Just one question. I could not agree with one point you raised, where you said it requires 4 liters of neem oil for coating of 1 ton of urea. I thought it was 0.6 kg of neem oil coating 1 ton of urea. Maybe I'm wrong, but I thought that was only one disconnect in the numbers and the presentation you gave.
It was the data.
Liters of oil. Please remember the government has granted the additional price of 250 rupees a ton for neem coated urea. If you use 4 liters of oil, almost 800 rupees, how is going to recover the money? So this is what is wrong. The second thing, it is related, since I asked the question, what is the source of your information when you quoted 88,000 tons of neem oil production in the country? The stalwarts like Sara Singhania, Dr. Venkatesh are sitting, they might tell me whether I am right or wrong. My information
That
says
was my
it is not
second
more
question.
than 15, 20,000 tons.
That was
of
my
neem.
second question. These numbers are a little off, sir. I would really like you to have a second look at the numbers. But otherwise, an excellent, leaving apart the numbers, the excellent work done by you for finding neem trees with 0.8 and 1.2 grams of aza per tons of neem, but that is fantastic. It may change the scenario altogether. And we really like to discuss with you off the stage how to go ahead and be part of the system.
And thank you very much for the questions. The data, it's not, it's only collected from the published resources. Probably I'll check with it and then.
Please do, because it doesn't match with what we know as a part of the industry. I'm sure you'll find a disconnect somewhere.
Yeah, sure.
Thank you.
This question, Dr. Kumaran again. My name is Venkatesh from AgriLife. Technical question. I didn't understand the word spray pollination. Can you just elucidate that?
As I said earlier, the issue, Neem has a issue of fertilization, pollination issues because of the self-incompatibility. And there's a lack of pollinators available in the plantations. So spray pollination is nothing but we collect pollen from the identified mother tree, superior genetic trees. And then we have a process of storing it. And then we try to spray the trees of the plantations. We can mix with some chemical liquid, and then we can directly spray to the flowers. It is happening in other crops. We've studied this, the entire technique from the experience of olive and one more species in Spain and all. It's published papers are there. Based on the scientific evidence, we started doing it, and there was a beautiful response in the plantations happening. It is a second-year trial. Probably will be standardizing the entire process in a year or two. Once it is done, it is going to be a biggest boon for increasing the productivity of the total neem seeds. Thank you.
Good morning, everyone. My question is again for Dr. Kumaran. Myself, Subhas Jha from Coromandel. Doctor, 2 questions. One thing you mentioned about neem being a cross-pollinated. Yesterday, I heard some of the speakers saying self-pollinated. So I'm confused there. So whether it's a self-pollinated or cross-pollinated, and if cross-pollinated, whether spraying some of the bee attractant will help, which is obvious from your presentation. So I need clarification there. Then the second point is you mentioned that neem extract business, global business is 2.3 billion or 2+ billion. I don't remember the exact number. So what are those extract which is taking it to that number? Because if I look at azadirachtin alone, it's 1/10 of that also on the higher side. And azadirachtin, as we know, is one of the major extract which is being exported. So that's my question and all. Thank you.
Your first question, Neem is a cross-pollinated. It is evidently done. We have done thorough work on that. Earlier, it was conceived as a self-pollinated species, because when you grow, it's a, say example, most of the trees are only scattered trees. We just thought that how come production is there? Then we thought that the neem pollen can travel a long time. And through our lab studies and then through our study, it's confirmed that it is cross-pollinated. And when you introduce local bees, honeybees, based population, there's a pollination and fertilization increases. So that's an evidence. And I already told it is a self incompatibility. Genogamus is not a 100% proven fact in Neem. So it is mostly cross-pollinated. For your second question, Neem extracts is not only azadirachtin, all extracts. Particularly the projection is because Neem not only in the agriculture, Neem extracts for the therapeutical and pharmaceutical industry is increasing like any time in the world. So it is a huge demand for, particularly for the pharmaceutical industries. There is a demand increase. So the Neem extracts includes all the extracts, not only azadirachtin. It's a common fact, data source. So it's not exclusively for azadirachtin. Thank you.
I think it needs more clarification because earlier studies at our institute, also by Dr. V.K. Gupta and others, he has studied the reproductive biology and he has told it is self-pollinated. So I think there is a lot of confusion is there and needs. Yeah.
I talked to him and we had the paper of him, and there is a misconception of the thing, and we approved it already. There are many other publications from other countries also. It is cross-pollinated. We saw in the field also. We could able to collect the pollens from the other trees also. Not only that, it accepts other Meliaceae pollen. Say, for example, Chloroxylon sweetenia, it accepts the pollen. Thank you.
Sir, I would like to add some more information on it. According to our earlier scientist has reported that Neem is a clistocami type of pollination. It means once the pollination happens, then the flower used to open. So that clearly ensures that self-pollination. Like Sir said, it needs some more clarity, some more clarifications that may be addressed. Thank you.
Good morning. I am Shibendu from ITC Life Science and Technology Center. I have a question to Dr. Gopinath. Sir, you have, in your presentation, you have showed that you have tried your semi-synthetic Neem compounds against 2 organisms. My question is, any particular reason you have chosen those 2 organisms, or it was just random?
No, see, as a chemist, we love to have structural activity relationship within the structure itself. So Neemine and Nimboloid. Okay, your question is related to the screening of the microbial. No, it was initial preliminary results. We are expanding. To start with, this is the strain we had with the collaborator.
Okay, thank you.
But we have intention to screen escape pathogens to go with AMR.
So if there is no any other questions, we conclude this session. Thank you all the speakers for this.
Now I request Dr. Arnachalam and Dr. Brar to come to the dais to felicitate the speakers. Dr. Vyas and Mr. Milan Mehta have taken the honor of felicitating the guests now. Thank you very much, sir. Now we can break for tea and reassemble at 11:15 for the next session. Thank you very much.
ठीक है ये ठीक है कल आपने फोटो नहीं दिया सर Yeah, we'll be talking about it. Thank you.
मेरा भी एक नहीं है
हाँ हो गया
और ready for show time? पर बालों के शो टाइम के लिए
है घर पे सैलून
ट्रेसेमे केराटिन स्मूथ केराटिन और आर्गन ऑयल फॉर्मूला फ्रिजी बाल दिखे सैलून स्मूथ 72 आवर्स तक इट्स शो टाइम ट्रेसेमे घर पे सैलून
This is Neem, the tree of 21st century. As a director indica AHS, it is one of the most suitable and valuable tree species native of tropical and subtropical regions. Today, it has been recognized as having the most potential of any tree of the globe due to its evergreen nature and ability to grow in even the most arid and nutrient deficient soils. Neem has found its glory worldwide due to its diverse range of actions and uses. Agriculture, human health, animal health, in almost every industry, the demand for neem products is growing. Of course, it is a boon for environment too. There is a huge market of neem-based products worldwide. Highest demand is in the pesticide and fertilizer industry. Over the last 30 years, the global price of neem fruits has gone up from 150 to 175 US dollars per ton to the current levels of 400 to 500 US dollars per ton. So we can say that neem industrialization has good potentiality. But what does that mean?
There is a big gap between supply and demand. After 2000, the popularity of neem has spread so wide. A day may come when Indians will not have neem for their own use. The amount of demand from abroad and all, it's growing so much. When I started the business, it was not such a popular product. But when days are moving ahead, after 90s, there was a demand for this neem. When Americans registered this product, they wanted to patent this product, and they popularized this product as a azadirachtin, which is an organic pesticide ingredient. Growth was very big. Actually, the demand was growing and they were using it. And wherever they used this product, they found it's a wonderful molecule. And in other countries, they could not get it. Though there are other plant products like pyrethrum and all, neem was very different from that. So this product has a lot of popularity and it's still growing. When they started doing this, the other countries like Vietnam, Japan, European countries, and Australia and all, they started using this product, and everybody found this more potent and very helpful product. So that is how I say when they see the market potential and the growth, the inquiries coming year by year and all, I feel that there is a big gap between supply and demand.
Today, almost every other product has neem extract in it. But the supply of these extracts, especially high quality extracts, is limited. To meet the demand of exponentially growing neem market, it is important to spread awareness. The benefits of neem are well known, but it is still not known by many how to obtain high quality and quantity of raw material. This is crucial because the whole chain of supply affects the final product being introduced to the consumer. Good quality final product means more sales, which lead to more demand. This is how any industry grows. Now is the time for Neem. In the recent times, there has been a major shift from chemical to biofertilizer and biopesticide. This has led to an increase in demand of neem oil. Though all the parts of neem tree are beneficial, neem fruits are the most precious as they are not found all year round. To meet commercial demand of neem oil, the neem fruits have to be handled with utmost care because it is the kernel of the neem seed which is filled with our liquid gold. The kernel is the richest source of oil as well as oleochemicals. So, we set out on a journey to find best practices from neem fruit collection to extraction of neem oil and other useful products. Neem fruit is a berry which is green when unripe. As the ripening progresses, the color of the outer skin, that is the pericarp, changes to yellow. On maturity, the fruit is fleshy and filled with sweetish fluid. It is estimated that a fully mature neem tree, which is approximately 8 to 10 years old, will yield 37 to 50 kilograms of fresh fruit, depending on the growing condition. Fresh fruit contains 40 to 50% moisture. So, 40 kilograms of fresh fruit will result in approximately 24 kilograms of dry fruit yield. Neem tree flowers from March to May in different parts of India. As a result, the fruits are mostly available from May to August. The collection of neem fruit in India ranges from meager 1% to maximum 29% of the total theoretical potential of collection. The primary reason for such poor collection are the unorganized plantations and unplanned collection effort. Neem trees are abundantly found in India, but they are scattered in forests, villages, semi-urban, and urban areas. Mostly, neem fruits are collected by tribals and locals who do not have enough knowledge and expertise on how to check and maintain the quality. As a result, the neem fruits collected have to undergo a series of segregation and rejection before reaching neem fruit buyers. There are two major solutions to this problem. Firstly, efforts should be made to educate tribals, farmers, and other locals so that they can be hired as trained neem fruit collectors?
We have to educate the people to collect the raw material the proper way. The raw material should be free from moisture, stones, sticks, everything. So we have to give the respect to the raw material. We are getting the traders from the North India. They are coming here. We have trained them to how you have to collect the material, how you have to buy the material from the farmers or local collectors or local other small traders like that. So now we are educating. Now it will be improved in future years, 2 or 3 years it will improve.
Secondly, organized neem plantations should be promoted so that standardized instructions for collection can be introduced and implemented. The main reason to implement harvesting practices is to ensure that neem seeds do not come in contact with the ground. This will ensure that the contamination of seeds by aflatoxin does not occur. Aflatoxins are a serious risk to the health of all users of contaminated products. Harvesting can be performed manually or mechanically. Ripened seeds can be removed by hand into a basket to ensure that no seed is wasted. Large clean tarpaulin, cloth, or layered net sheet should be laid out around the base of each tree being harvested so that any seeds which fall in the process are collected on the same.
You are late. There are no fruits now. However, I can demonstrate you how the women in our neighborhood gather fruits. We lay this net, which is at least 2 feet larger than the canopy, to ensure that all the fruits fall onto the net and not on the ground.
However, in some cases, like for manufacture of neem cake, even fruits or seeds collected
Good morning once again. Now I request the speakers for the next session, Mr. Madhivanan, Mr. Neeraj, Mr. Patil, and Mr. Gopal Sumi to come and occupy the dais. I request Dr. Kusum Arunachalam to chair the session along with Dr. Rajarajan, who will be the co-chair. And I request Mr. Dr. Abhishek to join me as the rapporteur. And before we start the session, I would like to honor Dr. Handa, who has been the chair for the previous session. And I request Mr. Vyas to come on the dais to honor Dr. Handa. The Each speaker will be allotted 15 minutes for the presentation, and the question and answer can be had at the end of the session. We have got 2 online presentations also. The first one will be from Mr. Lawrence. He will be presenting from Paraguay or from Spain? Germany, okay.
Okay. Good morning once again. And so I would like to first thank the organizers for giving me this opportunity at the last moment to chair this session. And I'm Professor Kusum Arnachalam, and I'm working at School of Environment and Natural Resources at Doon University in Dehradun, Uttarakhand. So it's nice to meet all of you who are working on this very important Neem tree, and being a part of this World Neem Conference is a great, it's a learning for me also to be here, and I have learned a lot of things. So now let's start the session. So we have the first presentation is from Lawrence Ward-Warbanke, and it's going to be an online presentation, and he'll be speaking on Nemobolite, a new technology to produce Nemobolite at scale and at an affordable price, a new offer for health and agriculture. So we'll request Lawrence to please go ahead with your presentation.
Yes. Good morning, everyone. Is my screen being shown? The presentation.
And you're audible. You can go ahead. Thank you.
Perfect. Thank you very much for the opportunity to speak here today. My name is Laurens van der Wülbecke, and you heard yesterday from my father about the Neem plantation in Paraguay and Pepe. I would like to talk a bit about some downstream applications, especially about Nimbolite and our research into producing more of it. As you all know, probably Nimbolite, and we heard some talks yesterday about it, is a molecule only found in the neem tree. It's primarily extracted from the neem leaves. And what we find very interesting is the rapid increase in research around this molecule. So moving further from whole leaf extracts into focusing on this molecule. So there is a lot of research around it, as can also be demonstrated in the World Neem Organization research newsletter, which is very good to read every month. We have a lot of mentions of Nimbolite, and there's a lot of research papers being published regularly about Nimbolite research. A lot of this is due to its interesting properties, which are mainly anti-cancer, antioxidant, anti-inflammatory, and antimicrobial and antiviral. We heard yesterday as well about some of these applications and some of the research already. I won't go into this too much. We've just shown in our own research as well, the antioxidant potential is extremely high, and the anti-inflammatory potential is really high. Anti-cancer, we would like to partner with researchers because we don't have the capability in-house. But the issue that we run across and that we talk with researchers about is that Nimbolite is expensive and unavailable, especially at scale. And we heard this yesterday in the evening as well. And we concur here that these are the prices. Yes, I know they are outrageously high, but as far as I know, we cannot buy this in larger scale. The largest quantity available is 1 gram. You can possibly buy this in larger quantities, but you cannot buy this at a kilogram scale. And I talked to one of these companies, Phytolab as well, and they told me that they are really happy if they produce 1 gram of it, and then they sell it in 1 milligram, 3 milligram units. So this is not something that is available in larger quantities, and this means that it also hinders research. So we talk to researchers in the cancer field, and they tell us that the availability and also the price of Nimbolite is indeed a problem, because researchers struggle nowadays with limited budgets. And if they have to spend part of it on Nimbolite, or if then they have to limit the amount of trials they can do or runs because they only have a limited amount, then this limits their research in a way. And there is a lot of interesting research going on in the research, especially in the cancer field. So we think this needs to scale up. But what is also an issue is on the budget side is that these researchers struggle with that the big pharma companies, especially, they are not so much interested in researching this. We all know about this. This is due to natural molecules not being able to patent it. So we, as the WNO, should think about how we can push this field forward, as one of the missions is to promote research on the Neem's potential for health. And the big companies won't do it because they can't make money off of it. So we need to think how to work together. But back to topic. So we with our company in Germany, Other Divine, we worked on a new combination of extraction and isolation processes to allow for the isolation of Nimbolite at larger scale, and then especially at a cheaper price. So this comes together quickly. As a Divine, we work only with leaf material. We get it from Paraguay, from NPP. And a lot of, especially the research on the extract side that we do was driven or is driven by consumer brands that we are also building up. So we have, especially in the cosmetics field. So we have the demand from the customers. We know what they want and how to formulate on the need for formulations. And we can put this back onto the research side and improve the extracts there. But what we've done now for the Nimbolite is a two-step process. We have our proprietary extraction method, taking neem leaves and producing a neem leaf extract out of it. This is, we use supercritical CO2 extraction. It has been developed over the last few years, and it has been especially optimized for high Nimbolite contact. We early on identified this as our target molecule, and we achieve a really high percentage of around 10% in our extract. And then in the second step, which we've later on researched now, is the isolation, which we go from this extract and we isolate the Nimbolite out in one single step process now. This allows us to keep the cost down and then also work with larger quantities. It's not easy because of the light sensitivity, the heat sensitivity of general different molecules in the leaf extract, but also of Nimbolite. But we've optimized this process now to work very reliably and in a very kind of simple way to achieve a 95% purity. So we're still working on scaling up 98% purity isolation out of this extract. We have it working. I'll show a graph later, but we want to optimize this for the whole process optimization to get the cost down, and continuously work on scaling up the batch size. But most applications are already fine with 95% purity. This is a graph from the lab very recently about for the 98% trials, 98.7 was achieved here. And this then leads to these two products that we can offer. The 95% purity in larger qualities can be certified as an analytical reference standard as well, and is available very soon. And then we're working on optimizing for the 98% variant for more demanding use cases, and this will be available later in the year. And that is my presentation. Thank you very much.
Well, thank you, Lauren, for the wonderful presentation. So we can take the question answer later on. So now I invite Mr. M. Mathivanan for his presentation. So he'll be speaking on enhancing nutrient availability, elevating stress level in leaves, and rejuvenating the dieback affected trees through integrated nutrient management in implantation. So if Dr. Babu can just introduce the speaker.
Yeah. Mr. Madhivanan is a PhD student at the Tamil Nadu Agricultural University, College of Forestry.
அனைவருக்கும் காலை வணக்கம். Good morning to all esteemed delegates who are all sitting on and off this dais. On and off the dais, I am Madhivanan from Tamil Nadu Agricultural University, Forest College and Research Institute. Actually, I feel very happy to present part of my research work with the topic of enhancing nutrient availability, alleviating stress levels in leaves, and rejuvenating dieback affected trees through integrated nutrient management. Next. I will start my presentation with a small outline. I will start my presentation with a small introduction and rationale why I choose this research work, and then methodology I have followed in this research and research I obtained from the research work, and finally my conclusion. Next. As we already know, Neem is the scientific name of Neem is Azadirachta indica. It's an evergreen tree, has its origin in the Indian subcontinent. It's called as Arista reliever of sickness because of its versatile utility. An estimation showed that India has about 25 millions of neem trees. Of the total, nearly 55.7% of neem trees was found in Uttar Pradesh, followed by Tamil Nadu, 17.8%, and Karnataka, 5.5%. It's a natural population. Nowadays, we are living in the era of sustainable farming. While we considering the sustainable farming, we should focus on how to utilize the biofertilizer to reduce the environmental degradation and how to improve the soil. And with improve the soil, we can maintain our plant health. Next. And this is my rationale why I choose this research work. As we already know that tea mosquito bug, it's a vector insect for spreading the dieback disease. So it take the Phomopsis azadirachta fungi, it will spread all over neem trees. So from our research work, nearly 100% of fruit was lost due to the dieback effect. So to overcome this problem, we utilize the effective biofertilizers for increasing the leaf nutrient content, and we also increase the stress level in the leaf. From this, we are maintaining the rehabilitation neem, and we improve the flowering also. Next. For my research, my experimental site was located in Tirunelveli district, Tamil Nadu. I used that age of my plantation is 4 years. Here I have used 4 biofertilizers with the name of Azotobacter, Phosphobacteria, VAM, and vermicompost and their combinations. Totally, I have 16 treatments including control. Next. After the application of biofertilizer, I measured the nutrient and biochemical concentrations like total nitrogen, total phosphorus, total potassium, and chlorophyll values, total carbohydrate, soluble protein, CN ratio, and the carotenoids from the leaves in the different stages. Then from that observations, from that nutrient and biochemical constitutions, I can derive the stress and the nutrient imbalance like CCI and CCR, chlorophyll content index, and chlorophyll to carotenoid ratio. And if I can know whether my fertilizer was effectively utilized by the plant. So I calculated nutrient source efficiency for nitrogen, phosphorus, potassium. Then I studied the dieback disease intensity. That dieback disease intensity was studied based on the score method. Here I fixed a score between 1 to 5, which the 1 denoted that there's a less amount of dieback intensity, 5 denoted that is a high amount of intensity. Here I used the SPSS software for my further analysis. Next. From my research work, there was a significant improvement while we are using biofertilizers. In my slide, it shows that when I upload the biofertilizer as a soil, all my treatment shows the significant value than the controlled. In my treatment 15 is the combination of all 4 biofertilizer. It shows the maximum total nitrogen, total phosphorus, and total potassium content. When we talk about the nitrogen is contributing more in green biomass and also flowering. If we noted the more nitrogen, it increase the leaf biomass, and the disadvantages, it will delay the flowering. Then phosphorus. Phosphorus also is a key element to increase the cell division and the formation of cell membrane. So it will increase the fruit development and this fruit initiation, the stage. It will use to complete all the stages. Then potassium. Potassium is it will break the dormancy and it will speed up all this process. When we talk about dieback recovery, these three nutrients are very important. First, the nitrogen will increase the biomass. While considering the flowering, that phosphorus and potassium plays a major role than the nitrogen because the nitrogen delay the flowering. So after the infection, we noticed that there was a huge amount of nitrogen present in the leaf because it is a stage of growth in vegetative biomass. After that, during the flowering stage, that nitrogen will reduce statically. Then that phosphorus potassium was increased. Then next. Like that NPK, that chlorophyll content also directly positively correlated with the green biomass, and it's used to recover speedy recovery of dieback disease. Here also that the treatment 15, that combination of all 4 biofertilizers increase the chlorophyll, all the chlorophyll values, then the control. Next. Then I measured the soluble protein, total carbohydrate, carbon CN ratio, then carotenoids. Here also the same results was I observed from my research work. That treatment 15, that combination of all will produce more concentration. Next. So from all those analyses, I can give some solutions. Then CCI and SSR. The CCI and SSR is positively correlated with the chlorophyll, nitrogen, and carotenoid. Once if it is increased, then that CCI and CCR, that its value range between negative to positive. Once if it is less than 1, so we have created some stress in leaf. So it will induce the tree to flower as much as earlier. Then nutrient use efficiency. Here I checked nutrient use efficiency, whether my biofertilizer was effectively utilized by the plant. Here it's proved that my all fertilizer was effectively used, because that value is more than 3, it will very good for leaf nutrient. Next. And then I studied the tree disease score. So as I already told, I give a number 1 to 5. So 1 means less intensity, 5 means more intensity. The tree applied with the combination of all 4 biofertilizers will receive the minimum amount of dieback disease and compared to the control. Next. So we also noticed that recovery time. As I already told, that 15th treatment will speed up the recovery. Then control exhibit, it's around 92 days. Actually, this is a major problem in Tamil Nadu. Tamil Nadu Neem growers are facing. So we are using this fertilizer to promote this. Next. And in my conclusion, to avoid this, in my conclusion, to avoid and to improve the nutrient availability in neem leaves, we are using biofertilizers. And at the same time, we are avoiding the dieback disease. And at the same time, we are increasing the flowering intensity also. So here, we are maintaining the sustainability with the help of biofertilizers. Next. And I would like to thank Coromandel International Private Limited for their funding, and I would like to thank Dr. K. Kumaran, Professor Farasi, who guided me to do this research work. Thank you. Thank you for all.
Thank you, Mr. Mathivanan. So now we'll move to the next speaker. We have Neeraj Dhakkar, and he'll be speaking on simultaneous detection and quantification of nimbolite and quercetin glycopanoside in the leaves of Azadirachta indica. So I'll request Balaji to kindly introduce the speaker.
Mr. Neeraj Dhakkar is a MSc student from Indian Agricultural Research Institute. He is a student who is doing his MSc in agricultural chemicals He is a student of Indian Agriculture Research Institute, New Delhi. He's doing
Thank
his
you, sir.
MSc in Agriculture Chemicals.
Thank you, sir. First of all, I would like to thank WNO for giving me this opportunity. Good morning, everyone. I'm Neeraj Thakur from MSc in IARI. So, from yesterday, all are talking about nimbolite and quercetin and other compounds, their beneficial effects. And previous presenter told about like separation of those compounds. So, here we try to quantify and detect those compounds. So, my presentation topic is simultaneous detection and quantification of nimbolite and quercetin in leaves of neem. Next. Is it working? So here, this is the morphology of neem. So already explained by other presenters. So I won't go in much detail. So every part of the neem has beneficial effects. So like it may be like seed kernel, bark, anything. So I won't go in much detail because it is already explained by other presenters. So so important phytoconstituents from neem are like azadirachtin, nimbin, nimbolite, and so now let's talk about my study. So objectives of the studies are, the first one is recently nimbolite and quercetin were identified as the major compound in the leaves of neem, and but no method has been reported for their simultaneous detection and quantification of both these compounds in the leaves of neem. And second one is the extraction of maximum amount of both compounds from the leaves of neem using different solvents. So the solvents we used were methanol and acetone. And third one was development of UFLC PDM method. So UFLC is ultra fast liquid chromatography method for simultaneous detection of both these compounds. So I will talk about some prominent bioactivity of nimbolite. So this is the structure of this compound, and so it has best property against potent activity against cancer. So it induces apoptosis in cancer cell lines, and second one, it inhibits cytotoxic autophagy in cancer cell lines, and in third one, it also like induces apoptosis in colon cancer cell lines. But no much work is done on insect cell property of this compound on insect. So I am working in MSc. I'm working on this Sporoptera frugipata insect. So it shows insecticidal activity against this insect, and other insects also has insecticidal activity like Aphanidermatum, Oxysporum, and Frazulina. And these are the benefit of second compounds. So this is the effect of this compound. So it also has cancer anti-cancer activity against cancer cell lines, and it is anti-allergen, and it has anticoagulant efficacy, and it increases bone density. So my focus was the estimation of both these compounds in neem leaves. So first we collected neem leaves, then we prepared extract using methanol and acetone separately. So here we took 1 gram of neem powder and then dissolved in 50 ml methanol and acetone. So then after that we concentrated it using rotary evaporator at standard pressure and temperature condition. And then after we kept it for drying for 1 day, so this was this we got. And then we prepared sample for UFLC analysis. As I told, it is ultra fast liquid chromatography. So here we prepared 1000 ppm solution, and then here it was identified and quantified using this UFLC PDA, and after that we got these peaks. So we optimized conditions for extraction. So at first, like I took 3 quantities of solvent, that was 25 ml methanol, 50 ml, and 75. So best was 50 ml. So 1 gram neem powder was dissolved in 50 ml methanol and 50 ml acetone. So here it shows that methanol extract has twice the yield as compared to acetone extract. As you can see, 0.23 gram per gram of powder. And this is for UFLC. So these are the standard conditions for UFLC. So here you can see the ratio of methanol and water is 70:30, and for flow rate was 0.9 ml per minute, and the wavelengths were used for 217 nm for nimbolite and 370 nm for quercetin, and the injection volume was 10 microliter, and temperature was kept 4 degrees Celsius. So these are the results. So an isocratic and precise UFLC method was developed for those two compounds, and it shows the method was showing linearity of R square. That is, you can see R square is equal to like 0.99, approximately 1. So for both standard cover. So this is the peak for standard nimbolite. So you can see peak, highest peak was for nimbolite, and second peak is for quercetin compound. And then you can say then we ran our extract in UFLC. So you can see nimbolide and the two peaks are overlapping. So it shows nimbolide is present in leaves. So we can quantify from this. And here quercetin is showing peak overlapping, two peaks are overlapping. So this also shows quercetin is present in neem, and we can quantify using the area under peak. So the first one was for methanol extract, and this is for acetone extract. So you can see here peaks are at not taller as compared to methanol extract. So as we can say, methanol has more higher peaks, so more higher yield as compared to acetone extract. So higher quantity of nimbolide and quercetin were present in methanol extract, as I told earlier. So here is the quantity. So methanol extract has quantity of 21.86 ppm, and quercetin has 32.47 ppm, while NEA has 21.58 ppm. So it is almost similar, but quercetin is very less. It is half as compared to methanol extract. So conclusion and future scope. So an optimum extraction protocol for maximum elution of both these compounds from neem leaves using methanol was developed, and then this method was developed for UFLC PDA, and then quantification was performed for both those extracts. And now we have to explore for other bioactivities, like for insect activities, because there is not much work done on these compounds for insects, and detailed characterization and mechanism pathways for insecticidal and pesticide purpose has to be explored in future. Thank you.
Thank you, Neeraj. So now we move to next presentation. So Mr. H.Y. Patil, and he'll be speaking on evaluation of Neem provinces in northern transitional zone of Karnataka. So I request Mr. Balaji to kindly introduce.
Dr. H.Y. Patil is a principal scientist in the Agroforestry Division of University of Agricultural Sciences, Dharwad.
Respected chair and co-chair, and on the dais and off the dais, and also the organizer of the WNO, giving me an opportunity to share my few views in the agroforestry, neem-based agroforestry system in Karnataka. Next, please. You know that neem is one of the important tree in India, and it's there is a genetic diversity also, and it has adaptability in wide range of climatic conditions as well as and the adaptive factors. It thrives very well in the dry and also the shallow soils, even though it's hard clay pan soils also. So that it grows naturally, even the 450 to 1200 mm rainfall, even though it is, comes up very well in the low rainfall areas also. So Karnataka is having nearly about 5.5%, and Uttar Pradesh is highest, and the Tamil Nadu is the 17%. So within the state, we have collected the 20 provinces in the year 1991, and we have to evaluate the performance of the different provinces of neem in Karnataka, and neem is our mandatory crop in the AICRP agroforestry. So that since it's environmental friendly, and it's neem-based pesticides have a low toxicity and for mammals and are common organic farming and applications. Next, please. Okay, you know that Karnataka is agroclimatic zones. It's a very diversified cropping system, it's having nearly about the 11 the climatic zones, 11 climatic zones, and also the diversified cropping system also. So among the 20 provinces, we have selected 4 provinces like the Raichur, Gulbarga, Bijapur, and Dharwad provinces. Next, please. So among the next, please. These are the 20 provinces. Next, please. Bijapur, Gulbarga, Raichur, Dharwad, and Dharwad provinces. You have to look into the crown area and average yield of the soybean crop and safflower yield. The Dharwad provinces was the more better as compared to the other provinces, as well as the Kharif in soybean and Rabi in safflower yield. The crown area is also the low in the Dharwad as compared to the other provinces. Next, please. Coming to the physiological parameters of soybean as influenced by the different Neem provinces, you can see that here the Bijapur provinces was the more the photosynthetic rate and also the leaf area index, as well as the SPAD and the chlorophyll content as compared to the other provinces. Next, please. These are the provinces. This is the Bijapur provinces. Next, please. Raichur provinces and Gulbarga provinces and the Dharwad provinces. Looking at the picture, you can see that the Dharwad provinces, you can see that the crown structure as well as the bole height of that the trees. Next, please. Coming to the seed and biochemical traits of Neem provinces. Coming to that, the seed yield and oil content, azotic content, compared to the other provinces, the Bijapur, the provinces was more oil content as well as the azotic content. That is the 41.26% of oil content and azotic is 1.24%. Next, please. In soil physical and properties of influenced by the Neem provinces, we have taken initial status of the soil in the water holding capacity and the moisture at field capacity. The initial is you can see that here 50% of water holding capacity and moisture is 28.6. For the years, as compared to the initial status to the initial to the cropping and provinces, the higher the water holding capacity and moisture at field capacity in all the provinces. Because of next, please. And also the soil chemical properties as influenced by the Neem provinces. You can see here the pH is there is no change in the pH as compared to the initial status, whereas in organic the content over the year it was changed. 0.48 to 0.66 over the year in the all provinces. Next, please. And major and minor nutrients like NPK also the change in the initial status also. As compared to that, there is no change in the NPK. Next, please. Secondary nutrients as also influenced by the provinces. The initial status of the soil is low as compared to that the combination with the Neem with the field crop is a little change in the little higher in the compared to the initial status of the soil. And these are the graphical parameters of soil physical and chemical properties. Next, please. Next. Coming to the economic evaluation of the Neem provinces under the agroforestry system, you can see here that the Bijapur, Dharwad, Gulbarga, and Raichur, compared to all the provinces, you have to get economic returns of the as a sole crop is nearly about 1.4%. And BCS is 2.46 in the Bijapur provinces. Looking to the provinces, the Bijapura is the one of the most high yielding, and also it's given the more grass returns as compared to other provinces. Yeah, when you compare to with the growth parameters, the other technical content and also the economic returns of we have to recommended the Bijapur provinces for further multiplication. To in the large scale large scale multiplication of the Bijapur provinces seedlings, we have to produce the quality planting materials of the Bijapur provinces to distribute the farmers. With this, I conclude my presentation. Thank you.
Thank you, Dr. Patil. So our next presentation is going to be online. So I'll request Mr. Hari S. Gaur, who is there online. So Dr. Gaur, can you first introduce yourself and then you start? So he's speaking on Neem management, Neem for management of plant parasitic nematodes in the horticultural crops. So kindly introduce yourself. Thank you.
My name is H.S. Gaur and I've been a nematologist all my life at the Indian Agricultural Research Institute of the ICR, and later on worked as Vice Chancellor at Meerut Agriculture University, and currently working as a distinguished professor at the Golgotia University. I've been teaching nematology, doing research, and also did work on Neem right from 1971 onwards. I've been working on Neem in various capacities. I was also a project coordinator of the All India Coordinated Project on nematodes, so monitoring research on Neem applications against nematodes. So I will share my presentation. Just a minute.
Sir, you share what?
Just a minute. Yeah. Yeah. Can you see it? Hello?
Yes, it is visible, but please make it full screen if you can make it full screen.
Okay. Right.
Yes.
Thank you. Is it okay?
Yeah, it's perfect.
Well, nematodes basically are organisms which are widespread in the nature. Any substrate you find nematodes, and they are very complex organisms metabolically and physiologically. But organization is quite simple. And they are pseudocoelomates, animals between the acoelomates and coelomates. They are vermiform, unsegmented, bisexual, bilaterally symmetrical, with well-developed organ systems, except specialized circulatory and respiratory systems. In fact, they are so numerous in the world that anywhere you find nematodes in all kinds of substrates. Plant parasitic nematodes, particularly with which we are concerned, they are the ones which are having these kind of stylettes, but they do cause diseases of man and other animals and all other kind of things. A few of them are also beneficial, which can be used as parasites of insects to control them, or the mollusks, pests, and others. Right. All kinds of crops, in fact, are affected by nematodes. You can see the vegetables or the cereals or the potato, or you can see soft crops, and also the hardy crops. Like pomegranate, you can see the decline of pomegranate due to root knot nematodes. They can cause yield losses and also deformities on underground as well as on the above ground plants. You can see the tuberose flower damaged by nematodes here. Losses basic due to nematodes are quite huge. About in India itself, so they say that nearly 65,000 crores are lost due to nematodes. Overall, 18% due to pests, and of this, 12% is due to nematodes out of this 18%. And you can say that overall, 2.6% of the entire crops are lost due to nematodes. In the world, there have been estimates of $157 billion worth of losses due to nematodes. life cycle is also quite complex. And because they live most of the time under the soil, you can't really see them easily. And many people don't know actually, and the symptoms are quite non-specific. They are symptoms like say stunting of the crops or the yellowing or chlorosis and everything, which is more often confused with the abiotic stresses and nutrient deficiencies and others. You can see the symptoms in patchy growths in the field, and the numbers are basically important. Where nematodes are less in number, crops are all right, they can be tolerated, but where the numbers are more, you find stunted growth like this. And the farmers are often confused. You can see all kinds of crops in protected cultivation and others. You can see a lot of damage occurs due to nematodes themselves, the stunting and yellowing of the crops, but also they combine with various kind of fungi, causing disease complexes. Nematodes cause injuries to the roots, and they basically allow the entry of bacteria and fungal pathogens into the root or the tissues and cause disease complex. Guava wilt, in fact, is a very emerging problem now. Melodogine and Tetralobi with the melodogine with Olsonia. You find this complex which is causing huge losses in the entire country. They can be endoparasitic, they can be semi-endoparasitic, they can be just outside the roots, ectoparasitic, not even realize that they are causing damage, and they could cause huge damage. Just because the audience may not be knowing what it is, so I just took some slides here. And there are various methods for manage them. Basically, there are many cultural methods for summer plowing, crop rotations, antagonistic crops. Some crops suppress nematodes, like marigolds, trap crops, destruction of infected crop residues, application of organic amendments. Here comes the use of neem also, where neem is an important organic amendment, neem seed cake particularly, but here it's cake. Use of resistant varieties and hybrids, physical methods like soil sterilization by heat or by steam, or by, and also biological methods, use of nematode antagonists, particularly fungi and bacteria. Many of them are suppressive of the nematodes. Botanical plant products, and among these, neem, karanj, and mahua. These three are Indian trees basically, have been very much used in Ayurveda as well as all kind of medical systems, Ayurvedic systems. And here comes the application in agriculture against various kind of pests, including nematodes. Work in India started in 1960s in Aligarh and ICAR-CAFRI and other places on use of these plants against nematodes, and particularly de-oiled cake was used as amendment. And the other parts also of the plant, they are also used for against nematodes. And you find various kind of reports in the literature. Thousands of papers are there used of nematodes against neem. There are chemical methods also, halogenated hydrocarbons or organophosphates, oxygen carbamates. Most of these are now banned actually, except a couple of them coming up in the market now. Neem in Indian agriculture has been, of course, very much used as a fertilizer, as a manure, for coating, neem-coated urea, you all know, for soil conditioning, and various kind of products basically. And also related plant, Melia ajadirak, is also Persian that is also having this kind of properties. So these two plants particularly have been very much used. Particularly extraction of neem oil from the seed kernels, that is very much used as neem extract or neem seed kernel powder or neem oil. Emulsions are made and which can be sprayed also, and can also be applied to soil and for nemato management, and also for root dips and other things. Chemical analysis has been done. Of course, claims were made about as a directant, particularly as an insecticide. And our research at IRA showed that more than azadirachtin, it was selenin basically, which was more related to nematicidal principles. But then there are various other compounds, niminin, nimicidine, or dihydro nemesidol or nemesinol or gadudin or selenine. They are all compounds which have been found in nematicide. There are a few others also. In fact, they are all, in all, there are total more than 40 compounds present in the neem. But when we see basically whether nematicidal action can be attributed to only one or two of these, we found that there are various compounds, in fact, it's a group of compounds which is acting against nematodes, and the nematicidal property could not be attributed to a single compound. And this we did a few trials on single compounds also, but then we found that it was a group of compounds which are explaining the toxicity or the suppression. Leaf, bark, root, seed kernel, powder, oil, and emulsions, de-oiled seed cakes. They are the different kind of, you can say all kind of parts of neem, even flower. They have been used against various pests, including nematodes. Seed treatment is done, seed coating can be done, or seed soaking can be done. Bare root dips have been done for the seedlings. Soil drains, spray applications against foliar nematodes, and soil amendment with the leaf or with the de-oiled seed cakes. Now, on these actually you can find hundreds of papers in India, and All India Coordinated Project on Nematodes had been doing multi-location trials all over the country, and all kind of departments, and there are various theses that come out of this. We have found, in fact, various kinds of actions on this, maybe biocidal, sometimes biostatic, basically. We found that low concentrations, the nematodes are not killed, but they became inactivated. So the action could be, it could be on the nervous system, or it could be on their other body systems, or the biocide. So multiple actions are there, maybe of different compounds put together. And the combined effect of all these compounds is basically more important. So products have been made from neem leaves, barks, roots, seed kernels, and they are present in market also. A lot of patents are there. The neem seed kernel powder extract, or particularly, or powder itself, they can be used for seed treatment and has been found effective in various trials. This provides protection for a short period, but then that is important to provide protection to the young plant. Neem oil is, as an emulsion, is also used for soil drench in nurseries particularly, and small areas. But then large areas, perhaps they cause limiting factors. Foliar sprays can be done against these foliar nematodes, and a few trials have been made on tuberose and others. De-oiled cake is perfect soil amendment. Basically, it provides nutrients, as serves as biofertilizers, and also suppresses nematodes, and supports various kinds of antagonists of nematodes. Many fungi and bacteria are supported because the carbon source is provided from this neem seed cake. So it has multiple actions. So that's why this is a preferred option particularly. And as a byproduct of neem, this can be used. But the problem is the large quantity is required actually. That's the problem. And the other problem is the variability. I'll come to that. So there are various advantages of neem over synthetic pesticides. They are biodegradable, they are natural products, they are multimolecular activity. That's why there's a lesser possibility of developing pest resistance. This is a problem we have been facing against various kind of nematicides, where nematicide resistance got reported after a few years' time. But neem, the advantage is that so many molecules are involved, and they're complex molecules. That developing resistance for the nematode is also difficult. So they're compatible with most of the other methods of management. That is important. And then for soil enrichment, and they're also cost effective if properly used in combination with other methods. There are certain disadvantages also, limitations you can say, the large quantities that are required. Stability is a factor that sometimes the old materials are used and they are not proven effective. And after a few months of photodegradability, other things happen. And the stability is a problem. And when storage is done improperly, the efficacy may be lost. There's a lot of variability in the different materials from different sources. We purchase from different markets and different places. Dr. Nutan Kaushik yesterday was presenting very nicely about the kind of variations that happen in the different components from different parts of the country. And this happens quite often. There's a lot of variability in the results does come. There are many patents basically have been found in Europe and other countries. You find a lot of patents have been done on Neem. And also many commercial products are now available in the market. I'm not advertising for any of them, but there are so many of them that have come in the market, and all kind of claims are made. And we feel that often good results are obtained, but even natural products from nature directly, the farmer is having access to the neem seed and or neem leaves particularly, and they should use it. And often we find that in the country, there are a lot of neem trees in every area of the country, but then you find that the leaves are lost. They are just dropped and lost, and also neem seed kernel is falls on the ground. But if some kind of scheme is made to encourage people to collect them and to market them, this can become a good earning or secondary source of income for many of these people. Efficacy is also based on the different kind of nematode species, pest species, soil conditions, also application methods. So standardization is required, particularly and to define again how much is required against what species. That is important. Some of the nematicide molecules may not be stable after when you take out of the plant, and that is a problem. So appropriate storage conditions are necessary to maintain the efficacy of various active ingredients. There is variability in the concentrations and efficacies, as I already said. And in different ecological conditions, that's what they determine these contents, and also the genetic diversity in the neem itself. Standardization, therefore, is necessary. But the present day, the standardization is mostly done in the directing content basis. So far, as far as I know, I've seen a few reports on standardization based on selenium and other things also. In 2023, I found a paper. And so basically, we need to know for nematicide action which compounds are more relevant and what should be the purpose of standardization. If against neem, against nematodes, then what should be used? Against insects, what should be used? Neem products and neem-coated impregnated products have been found to suppress.
Sorry, Dr.
root
God,
knot
to
nematodes.
interrupt you.
There is,
You can please
yeah, just finish.
wind up in 2 minutes. Thank
Yeah,
you.
just winding up. Okay. The various kind of reports are there, and just cited a few here. Few synergistic reports are there, particularly against the purpose of linacenum, Oconia clomatosporia, they are biological control agents of neem against nematodes. And these can go along with neem actually, but they are not suppressed. So they're compatible also with the entomopathogenic nematodes. When you apply to soil, the nematodes, EPNs are also present, but the reports say that they are not killing the bacterial feeding or say entomopathogenic nematodes. And although there is some variability, one or two reports show that some nematodes are suppressed, like Heteroblaitis indica. One report says that it was suppressed. So it has to be confirmed whether they are doing or not. So nematode the use of neem also has to be a part of overall IPM as a botanical pesticide. It cannot be a standalone management, but as a one component of IPM, it serves very well. So this is the last slide. I'll just conclude. More research is needed on the mode of action of these different compounds. Synergism is also to be studied, and compatibility aspects to be confirmed before recommending applications. New facets of neem's nematicidal properties have to be explored. More work has been done on insects, but very little on nematodes as far as basic research is concerned. Innovative formulations, particularly to reduce quantities like nano formulations, can be tried. Storage conditions and shelf life are to be standardized. Integrated neem into broader sustainable agriculture practices. That is the goal basically. But one caution I will just say, although Neem has multiple uses and various claims are made, but we should not, I think, perhaps consider Neem as a panacea against everything. And I have seen some tall claims also made in literature many times. So we have to be cautious that although it's very important that it has multiple uses, but we have to be cautious before making a claim. Thank you very much. In fact, my acknowledgements to Google Images from here. I've taken some of the pictures. Gloria University, MIT University, and Professor Nutan Kaushik particularly, who insisted I should make this presentation on nematodes, and I think this is the only one on nematodes in this conference. Thank you very much. Thank you.
Thank you. Thank you so much, Dr. Gaur. So we can have questions from the audience because you are online. So I'll request the audience if they have any question, please, you can ask.
The questions will pertain to only this
this
presentation.
one.
Right.
Mike.
No, he's bringing the mic. He's bringing.
Yeah, the mic is coming. Just please wait.
This one.
I've been personally out of touch for many years, actually. Maybe some more recent things have come up I might not have covered, but my interest does remain in him still.
Very good, doctor.
Excellent, a very informative talk. So
That's all.
Dr. Gaur, I have one
Yes.
question. During
Yes.
your what you call All India coordinated tries, when you
Yeah.
tried the Neem for the nematode control, what was the control
Yeah.
which you could up to what percentage you could manage on
Nearly 50 to 60%. Well, that's why I said there is a lot of variability. Sometimes we will claim 20% control, but some are claiming up to 80% control. 50% control is provided, and that is in fact a very good level of control. In fact, even nematicides are not able to give us full control. Nematicides also they gave only 70-80% control. So even if Neem gives out 50% also, there's a good level of control. And only our aim is to reduce nematode populations below the tolerance level of the plants. So they are not able to control 100%, but on an average, I can say around 50 to 60% control can be obtained depending upon the dose and the conditions.
Thank you.
any other questions for Dr. Gaur?
Yes. This is Joe from Tamil Nadu. My question is, Doctor, you were saying about cultural practices to control this thing, nematodes.
Yeah.
And one point you said is use of resistant varieties and hybrids.
Yeah.
Do we have any variety or hybrids in Neem? Resistant.
Not Neem. About the crops we have against nematodes, we have hybrids which are resistant to nematodes, a few of them, and also some resistant varieties, also available in tomato and potato and some other crops. So some resistant varieties are already available. In wheat and barley also there are some varieties available which are resistant to nematodes, but there are not many actually, and still there is a paucity of resistant varieties against nematodes. But I have no knowledge about the hybrids in Neem actually. I don't know. There may be many. I'm not talking about that. So I was talking about resistant varieties of crops against nematodes.
Yes. Thank you, Doctor. In continuation, another small question. That is, in some places in our district, that is Tirunelveli in Tamil Nadu, we have a peculiar type of variety probably, and it grows well, very fleshy growth is seen during summer. And during winter, we see the plant dying, and I mean, totally something like dieback. And again, when the summer arises, starts, it comes well. So can you find any reason for that, or will you say it is a separate variety or things like that, species?
No, you're talking about which plant you are talking about? You're talking
I am talking about neem.
Neem, actually, it may not be the effect of nematodes directly, although nematodes do parasitize neem also. But I have not seen the effect of nematodes on neem. What you are saying, perhaps it may be nematodes are also involved because neem itself has its own quota of pests and diseases, but nematodes may also damage to some extent. And in fact, when the new season comes, the spring season comes, but new leaves do come actually. There is a general decline in crops. Any crop, any particular tree, if the pests are damaging the roots, the growth, the recovery after every season may become less, and the overall longevity of the tree may be reduced. But I'm not really sure. I don't have direct data on what you are saying.
Okay, doctor. Thank you. Thank you. I think.
Thank you, Dr. Gaur, for the excellent presentation. And now we'll one more question is there? Or? Okay. Please listen.
Thank you. Thank you very much. Then.
Dr. Gaur, one more question. Sorry for that.
Yeah, please. Yes.
I just want to know, you said there was a synergistic effect with Pachonia, and then you said selenin was the most active compound, which was isolated from leaf. Leaf or root?
No, we in fact got the molecule from the Agricultural Chemical Division of the IRA. I don't know from where they're taken, but we got selenin from there. And we tested that. In fact, my colleague, Dr. Vijayalakshmi Majumdar, late Majumdar, she was doing a lot of work on this, and I was associated with that work. So we found that, in fact, I myself tried even 1000 ppm of azathioprine directly, not killing nematode in the suspension, but selenin was killing.
Yes, sir. So selenin with this Pachonia, if it is working, it will be a good product.
No,
right?
Pachonia, because Pachonia directly when you apply, it is having some nematode suppressive effect. It's a biological control agent. But then if you apply along with neem, it was more effective. That is why we said it's synergistic. And perhaps it was because of the carbon source provided by neem, and the Pachonia itself was thriving better compared to in the absence of this organic matter.
Sorry, in continuation to this question, Dr. Gaur, so what was the result of the selenin alone? Oh.
No, selenin alone was killing.
Killing.
Selenin alone was killing. In nearly 24 hours, the nematode was first immobilized and then killed. Azathioprine also, after 24 hours, we found some inactivation, but we found selenin was more effective compared to azathioprine.
Thank you.
And there's a paper by Vijayalakshmi Majumdar on this. Thank you.
Okay, I think if people want to know more, so they can directly connect to Dr. Gaur. And now we are running short of time, so we'll.
Thank you. Thank you. Thank you. Thank you.
Lawrence, if Lawrence is there, we can have question for him also. Yes, any question for Lawrence from the audience? If no question, then we'll move to the last presenter of this session, and I'll request Mr. Gopal Sami to please present his talk, and he'll be speaking on Neem Seed Strategy 2024. Dr. Balaji, if you can just.
Mr. Gopal Sami represents KGS Corporation in Chennai, and then he will present on the Neem Seed Strategy. Thank you.
My name is Gopal Sami. I came from village level in 1975. In the school days, Saturday, Sunday, I go to village. I collect the neem seed, castor seed. At the time, 10 paisa per kg in 1975. Then I keep it in my house. After 10 days, I go to nearest mandi. I will sell the raw material. Then whatever profit will come now, I keep it for my studying purpose. That is my life started. In 1990, I joined in Tata, first Tata Oil Mills. for the neem seed procurement. Then I moved to EAD Perry, 1995, head of the neem seed procurement. At the time, only neem fruit only come to market. Lot of neem fruit only has come to market. Then each and every village, I will go there. I will convert it to neem fruit to neem seed. One of the some traders, sir, you don't go, no work, please go. It is not easy. It is very tough job. Neem fruit converted, neem seed, it is not very easy. I told him, please sit, I will take care of. Then I put a lot of G campaign every week in the 2 months of the neem season. Lot of G campaign. Slowly I converted neem fruit to neem seed. Neem seed in the market. Up to actually in 1990, before that, all the neem seed, neem fruit go to cake and oil manufacturers only. Now after 1990, that we found that technical azadirachtin, then only changed this neem business. Everybody knows after 1990 only. Second thing, this is the South India, Tamil Nadu, Karnataka, Andhra is the highest asa content in India. Very good asa content in the India. In north side, in MP, this is somewhat 0.3, 0.25 asa content. It is not suitable, not viable for the asa purpose. So everybody wants good quality of neem seed, more than 0.5 asa. So only Tamil Nadu and Mysore and Andhra, some part only, it is more than 0.5 asa content was available. So we concentration more. I have done more than 25 years, only asa only. I can easily tell how much asa content in the seed without HPLC. I can tell how much asa content in the seed. Before that, we send a sample to factory. HPLC take more than 24 hours. The season only 45 days. We can't achieve the quality. So then, based on the experience, I break the kernel. I can tell how much asa content, which area, which is the neem seed, which area belongs, everything I can tell within 5 minutes. It is very easy for procurement. Next slide. Next. Introduction of value of neem. I worked in Tata Oil Mills Company, them neem seed procurement since 1992 and EAD period up to 20 years. During 1990, neem seed was less available, neem fruit was more available in the market. Neem cake and neem oil was primarily produced for the cake and oil. Neem produced for the neem seed and fruits before 1990. Neem cake was primarily used for organic fertilizer, while neem oil used for the natural soaps. After 1990, research breakthrough from the foreign countries led the invention neem technical powder azadirachtin from neem seed. Then major companies focused the raw material procurement. Gradually, amount of companies increased one by one, like PG Margo, EAD Perry, Fortune Biotech, and Ozone Biotech, and others, more companies. Next slide. Some of the companies would not sustain in the business because of their inability to produce the asa due to various reasons, including method of process resulting instability of the technical. So many companies in 90 up to 2000 ITC, they speak, so many companies has entered in the business, but they closed due to inability of the technical process. EAD per created factory to produce asa. The demand for the raw material was close to 10,000 tons per year. We are able to run the factory only 40 to 50% capacity because of lack of demand. During this phase, we also reached out rural South India, Tamil Nadu, Karnataka, AP, and educated the neem seed pickers to get the right amount and quality of raw material from fruit to neem seed. Market demand of also increased from 5,000 tons to 20,000 tons between 1995 to 2017, as the market understood the quality of ASA technical organic farming all over the world. During 27 years period, there was no issue in the supplying the raw material of ASA production, as well as the cost of production and sales did not change. Then, in 90 to 2017, 1 ton of neem seed, we get 1.6 kg of ASA technical 1 ton of seed, 1000 kg of seed, we get 1.6 kg of ASA up to 2017. After 2018 to 2024, now 1 ton of seed, we are getting 1 kg of ASA due to adulteration only. In reason for the change the raw material output, I bowed the way since 1995 to 2000 for promotion of raw material from 5,000 to 20,000 tons, picker concentration more on the quality of raw material than no major change in the climate condition. I left the company 2011, 6 years till 2017, the foundation was built smooth and stable. After 2018, there was a huge competition of raw material due to high demand of ASA powder and the neem oil for urea coating, leading to scarcity of raw material. Reason for the scarcity: lot of neem trees were cut because of the road extension in the rural area, change the climate condition after Corona, boom the industries led to manpower scarcity. Due to unhealthy competition to meet our heavy demand, industries were not able to meet quality standard of raw material. So ASA recovery has come down more than 30 to 40%, and simultaneously price increased 4 to 5 times abnormally. Next slide. You see the neem seed democracy and availability. In Mysore, more than 2,000 tons, 2,500 tons up to 2017. After 2017, it's come down to 1,000 tons. You see, each and every crop, every place. Salem, more than 1,500 tons come. Today, only 200 tons only is coming. More than 50% arrivals has come down. You see, Gauriputtur and Tumkur, Kadri Andhra, Karur and Utangarai, Sangam, Velur and Tirupattur, Dindikal and Harur, Annur near Coimbatore, Krishnagiri, Pannagaram, Vyapanapalli. The highest ASA content in India, in the world. Number 1 is a Pannagaram, the Dharmaburi district. I can challenge today also. It is my baby. I know how much ASA content in the each and every place. This all the market I developed. And Kolpatti, more than 2,000 tons, 3,000 tons seed will get. The Singaman area and Pudukkottai, Tamil Nadu area. But ASA content is very less than 0.4. It is not stable in the seed also. That is ASA drop. The Pannagaram, the Salem, Mysore, this area and the Dindikal area, the ASA drop, it is minimum, very less ASA drop. Except other area, this Kolpatti is very fast, and Pudukkottai, Singaman area, Tamil Nadu area, ASA content very drop. Even though you keep it 1 month also, ASA content will be dropped. That is due to the quality of the raw seed and sand, the area. You see, next slide, sir. ASA content, next, you see, next, sir. It is Tamil Nadu, next, sir. ASA content, yeah. You see, Mysore, 0.7 ASA content in the 2000, today 0.45. And Salem before that 0.65 ASA content, today 0.6, less than 0.6. In Gauriputtur, Tumkur, it is more than 0.65 ASA content, today 0.4. There is no market in the Gauriputtur, Tumkur. It is gone. There no, even though single seed not come from this Gauriputtur, Tumkur area. What happened? Due to competition, everybody killed that area. Gauriputtur very good, Tumkur very good Asa content in up to 2000. Now, due to competition, lot of competition, there is single seed not are coming from the, and Kadri AP, highest Asa content 0.65, today less than 0.5. Karur and Utangare, you see, more than 0.75 Asa content, today 0.5. Sangam, Velur, Tirupattur, this is all are more than 0.7. This area, in Tirupattur, Velur area, more than 3,000 tons I procured 2010. Today, 300 tons only come to market. What happened? Only God only knows. Lot of neem trees cut, manpower shortage, climate condition, so everything the Minkil. The Vapanapalli, Pannagaram, Krishnagiri, that is more than 0.7 Asa content. 1 or 2 bags it come to, Dr. Kumaran sir told me more than 0.8, 0.9, only 1 kg or 10 kg seed. If you go to bulk, more than 200 tons, 300 tons, 400 tons, it immediately, we can't bring to factory, we can't Asa extract immediately. That is why it is 0.8 Asa, slowly is coming down 0.7, 0.75 average. That Teni and area 0.5. In Kolpatti, this Pudukkottai, the Virudhunagar, Manappalli, South Tamil Nadu, that is 0.4 only, less than 0.4. This, all the material, this all the material coming to high Asa area, it take it.
Mr. Gopal Sami, please wind up in 2 minutes.
Yeah, what, no, over, last slide. Yeah, that is why Asa content, this Kolpatti area, this area, gone to, they are mixing with high area, it is 0.75, 0.7 Asa, it come down to less than 0.4. That is the scenario, sir. Yeah. Yeah, last slide. Yeah. My role is the January. I joined February 1990. I increased gradually 20,000 ton, 2011. My challenge was due to identify right quality of seed using HPLC machine and get report from the labs used to take close to 3 days time against raw material availability window, which was only 30 to 50 days. I developed a skill, ascertain quality of the match report looking in the seed instantly to avoid the delay of the procurement. I was also named mobile HPLC due to this by Americans. I can cite kernel and determine the amount of Asa content in the raw material. That's all. I am also producing neem cake, neem oil, neem concentration, neem technical powder. I am also producing 15 to 40%, 15% purity technical, 40% purity. Any more questions, please ask me.
Okay, now the session is open for question and answers. So
May I ask one question to?
Yeah.
Did you correlate this as a drop with some kind of climatic conditions, like temperature or humidity or the rainfall?
Which some area, you take it, Asa will not drop. In Pannagaram, even the keep 6 months, 7 months also. Today also I'm keeping the material. 0.7 means 0.65. But in this South Tamil Nadu, that is Kolpatti, Singamaneri, Asa drop very fast due to climate condition.
You showed in the one slide in different years.
Yeah.
In different years, Asa is dropping.
Yeah.
At one point of
Not
time,
year,
it was,
only 5 months only.
but you said 2011, 17, 2018, 18, 23.
Yeah, Asa dropped due to.
Yeah.
Asa, same Asa is there. This adultation, this South Tamil Nadu is highest Asa content in this Kolpatti area. This material, they are mixing with 0.7 Asa. It coming to 0.45. Okay. Only adultation only.
Okay.
Asa will not drop. Even though today also 0.7 Asa is there. Mainly they are doing adultation.
Okay. So, but it was not clear from the side.
Yeah. Only adultation, but I can't tell you everything, no. Tell me, sir.
Thank you, Dr. Gopal Sami. It was a wonderful presentation. I would like to say here that I have been seeing all presentations from since yesterday. It was the first presentation regarding neem seed. We are working on the neem summit. And if there is no neem seed, I think there won't be any Neem summit. So we have to, as you have mentioned very clearly, that we have to procure, we have to save our Neem trees, we have to save the Neem seed. So I would first of all congratulate you for this, and again I'll request all the office bearers of Neem Summit that if we can hold of this kind of any meeting, because you are the pioneer and so many persons from South and Rajasthan and MP have come here, even I'm from Uttarakhand. So if we can do something for the saving of the Neem trees, saving of the quantity of Neem tree, quality of the Neem, and the ASA also content, what you have said. So it would be better for the health of total Neem industry. Thank you.
Any other question to the other speakers? So
I have one question to Mr. Patil. You have shown that the net return profit for the farm, its kind of profit is per hectare per year is 10,000 rupees.
Yeah, madam.
Is it from the block plantation or it's from the bud plantation?
It's a block plantation, madam.
Block plantation.
8 by 4 meter spacing.
So do you think this is sufficient for the farmers to grow the tree, the neem tree? This
Pardon?
10,000 rupees per hectare
Yeah.
per year amount is good for the, is attractive enough for the farmer to grow the neem tree?
Yeah, earlier, madam, first of all, the 6 years, 10 years, there is no changes in the yield of the agriculture crops. After that, that will reduce the agriculture yield.
No, my question is that, like suppose I am the farmer, I want to grow the crop, okay?
Okay.
I want to grow for some economic returns, no?
Yeah.
Not for the charity or something.
Yeah.
So if I get only 10,000 rupees in a year from 1 hectare, will I be interested to grow the neem tree?
You have end products from the timber as well as the agriculture crops.
I don't think.
Pardon?
we can move to the next question.
Hello. Doctor, you can have this discussion. You can have the discussion offline with them after the session is over.
Good afternoon. My question is to Gopal Swami and H.Y. Patil. I am Dr. Yadav from IFCO, and IFCO has developed 6 varieties of neem with the collaboration of FRI. Actually, I'm listening from yesterday. There are a lot of things industry and uses are going on. But I ask what initiatives you have took to make plantations, huge plantations, because everybody is saying material is not coming. So new plantation, for the new plantation, what is the encouragement to the farmer? As rightly Madam has said, if farmer is taking only 10,000 rupees, so whether they will be ready to make more plantations? So what will be the strategy to increase more and more plants at the farmer field? So that is my question.
In my experience, so many lot of neem plantation in the India, in Tamil Nadu, but it is natural means natural. It is even though Perry more than 600 acres, Indo-Burma trading Bombay 1,200 acres neem plantation in near Tirunelveli. We didn't get even single kg of seed in the plantation. I do not know what is the reason. I think it is natural means natural only. If we get more manpower, the cost of the neem seed collection per kilo, they will it more than 200 rupees will come. We put manpower, they will collect morning 2 hours, they will work. They will and evening they will work 2 hours. We want to give the coffee, tea, lunch, everything. It is very tough. It is natural means, in my experience, natural means natural. And we can give to government, we'll inform to government. Otherwise, we put Jeep campaign, some advertisement in the 2-month area, we'll spend more than 50 lakhs. It is good. Otherwise, will come to more market. Tell me, sir.
I have a question.
So thank you. Looking all these problems, actually, IFCO has again entered a MOU with the FRI to develop a dwarf variety of neem, because collection is a major issue.
Yes.
So hopefully within 6 years, we will be in position to develop a dwarf variety that will be a game changer for the neem industry. Thank you so much.
Mathivanan, see, you spoke about enhancing nutrient availability, elevating stress in leaves, and regenerating dieback using integrated nutrient management with biofertilizers, right?
Yes, sir.
So, have you ever studied on the impact of this biofertilizer in plants in terms of reducing the incidence of tea mosquito bug incidents by enhancing the cell wall reinforcement? Because this NPK consortium is known to do the cell wall reinforcement in the plant. So there is a strong correlation between the pest incidence and the sturdiness of the cell.
So actually, I measured after the affection of the tea mosquito bug only, sir. So
The
actually,
subsequent incidents, have you ever studied whether there is any reduction in it or any reduction in the dieback disease?
I didn't study, sir, but we studied the biofertilizer, then how it will recover the dieback disease and how it will promote the flowering, because once the dieback will affect it, then flowering will reduce. So then how it will decrease the dieback and increase the flowering.
No, my question is to you only. Do you recommend that if we do the proper nutrient management, the dieback disease can be managed?
Yeah, ma'am, we can manage the dieback disease after the affection. We can
After the, why not before?
actually, I did my research work in.
No, forget about your research work. I'm just in general. Do you think that
Yeah, ma'am, we can
do that kind-
arrest. We can arrest the if we control the insect, we can arrest the dieback effect.
Oh, it's a insect-induced dieback. Okay.
Yeah, ma'am. Actually, there is an insect called tea mosquito bug.
No,
It's
please, tea mosquito, but
it's act as a vector, ma'am.
so how this nutrient management is helping this?
Ma'am, that nutrient will help after the affection, ma'am. How to rejuvenating.
Okay, that's only to fight back. Okay, fight back again. For the new growth, okay, nothing.
Okay, thank you.
I have, sorry, one question
Okay.
to anyone who wishes to answer. As a farmer, we have learned the cost of land in India is exorbitant. So just want to know, is there any kind of government subsidy for farmers to procure land for the cultivation of neem? And if not, are there any collaborations between major producers who have the demand for the neem seed or the leaf to partner with and adopt local farmers and support them so that they can guarantee their product?
In the previous lecture, I think Dr. Kumaran was mentioning about a collaboration wherein there is a contract forming of Neem being taken up in collaboration with one of the industries in Tamil Nadu. I think you can discuss with Dr. Kumaran more about this collaborative way in which this arrangement works with the company as well as with the farmer.
But is there any government subsidy provided?
For the land procurement, there is no government subsidy, I think. I think that's a policy decision which I may not be aware of. If there is any collaboration with a company or with a farmer, it's only a bipartite agreement which they go on with that. Government doesn't provide any free land or subsidized land for the growth of any crop or any plantation. If I'm right or if I'm wrong, and others can substantiate this claim.
Hello, sir. Myself Ajay Bharadwaj from Gramin India, Indore, Madhya Pradesh. My question is for Gopal Swami, sir. Actually, we are in same domain. I'm also collecting the raw material. We are facing the problem of horse trading, like what quality material should be gone on quality prices and non-quality material should be in low prices. But in our area, what is happening? The quality material is not getting the quality prices and the non-quality material is trading on high prices. So what should we do in this regard? First.
Your area material?
Sir, actually, you are working in Tamil Nadu.
Yes.
And I think you guys are taking step on non-quality material trading should be stopped on high pricing.
Where it in your area?
We are in our area. The basic problem of ours is that
Your area Assam content is very less there. It is not suitable for the Assam manufacturer.
And the second question is, first question, we are doing for stop this horse trading in our raw material market. And the second question is, what kind of storage practices should we take in during collection time so that ASA could not be dropped?
So even though whatever steps you take in MP or this area, it is ASA is the climate condition, due to climate condition, ASA's content is just 0.3, less than 0.35. It is very, in Tamil Nadu, all the neem fruit, automatically it will fall down the ground floor. But your area, this North India, dried, in the tree itself dried. That is why ASA content is and very hot also, and more than 45 degrees Celsius in summer. In Tamil Nadu,
he wants to know how to preserve the seed so that the ASA doesn't fall.
they are giving the neem fruit.
That's what your question.
Yes, sir.
Yeah.
Your area, it is
Sir, we have a problem that the collection time and the pre-monsoon time the same.
Yeah, same.
So, we just wanted to procure a quality material so that if it is
you keep the dry immediately
for ASA also.
Immediately dry the material.
Yes, sir.
But your people are not drying.
Okay.
In Tamil Nadu, immediately they dry.
We are trying to dry in dry.
No, immediately you want to load the material, but it is 4 to 5 days to transit the ASA content is coming down. Immediately dry the material. At least you can save the more than 50%. You can save the ASA content.
I think we discussed during the lunch, so now we will break for the lunch.
My request is that we are into collection, basic collection, guys, and we just wanted to educate the people who are downline working. So I just wanted to your guidance for collection, and we should educate the lower people so that the quality material should be collected, sir. Thank you so much.
Thank you.
You can have other questions also.
Okay. I have one question. My name is Asit Shah from Organics. I'm here on this site, Nairobi, Kenya. This is for Mr. Gopal Sami. Now, this is a very interesting thing that you're doing about your working HPLC. Now, is this a skill that you can pass on to somebody? Are you able to teach people how to do this? Because I think it's something which can be done really fast. And I think the skill should not be lost. Have you trained anybody to do this? Is it easy to do it? Can you tell us a bit more about it?
It is correct, sir. It is based on the experience. Now today, in 90 up to 2015, only myself educated, only carbide people. Now everybody knows, 50% people know how much ASA content. I educated so many suppliers, how much ASA content. If you see the seed, I can tell how much ASA content. Then the same thing I educated, but they want to sell the raw material. That's all only. It is a very tough sum, but experience based on the experience only. More than 20 years experience, each and every area. That is based on the experience only. I got this input.
So you have other people who have the same experience, and you've been able to teach them.
Here in Tamil Nadu.
Okay. So I think, is there some way of taking photographs showing this is a good quality seed, this is as a content?
Yeah, I can tell easily.
Okay, so last question.
This is not question. This is land availability and partnership. We have a land in Madhya Pradesh and Rajasthan. We have an agreement with the local panchayats, and in Rajasthan we have around 5,000 hectares land available for plantations. We are also doing plantations, but if somebody wants to join hands, particularly for neem plantations, they are welcome. Madhya Pradesh and Rajasthan. Thank you so much.
So,
and whereas our research organizations like we are having large neem plantations, and we are facing the problem to how for the neem seed. We are putting tender also, but not getting the buyers. So there is need to have a more collaboration between these.
I
Just
think,
like-
yeah, this can be documented through
Yeah.
this recommendation which will come out of this. Thank you. Okay, so we would like to thank all the speakers for their wonderful presentations and the participant for a very informative discussion which we had. So now we are breaking for the lunch, and after that we'll again.
I have few announcements to make. Now the poster session is open. All of you can visit the poster session and view the posters. Secondly, the lunch will be available from 2 o'clock onwards, and the valedictory function will start at 2:45 in the same hall. I request all of you to kindly visit the poster session and encourage the students and scientists there. Thank you. And before we break, I request Mr. Vyas and Dr. Venkatesh Devanur to come on the dais and felicitate the chair, co-chair, speaker.
Please remain seated. Please remain seated. Just 2 more minutes. Please remain seated. Friends, in the award ceremony from WNO, yesterday there was award for Dr. Baral from Chittaranjan Institute, Canada, for the eminent, outstanding scientist who has done the great work for Neem. Dr. Baral was not there yesterday, but today he is amongst us, and we would like to honor him. I request Dr. Baral to come to the stage, please.
WNO is very happy and honored to award the Outstanding Neem Scientist Award to Dr. Baral. Yeah. Dr. Baral of Chittaranjan National Cancer Research Institute, Kolkata. He was born in July 03, 1962, and has nearly 30 years of experience in cancer-related research. He is a postdoctoral scholar of Markey Cancer Center, University of Kentucky and Lexington. Presently, he is the head of Department of Immunoregulation, Immunodiagnostics at the Chittaranjan Cancer Institute, Kolkata. His major field of research is on immunomodulation in cancer prevention and treatment. Dr. Baral is a member of several professional societies. He has got 4 book chapters, 3 reviews, 54 articles, and 1 patent to his credit. He has supervised a number of PhDs and even many MScs and various short-term projects. WNO is very honored to award this Outstanding Neem Scientist Award to Dr. Ramindranath Baral.
I am very much honored to receive this award. I can say from the beginning of INO, WNO, so I am associated with this, and I am a Neem lover and Neem researcher, and this is a good platform of us that is the Neem lovers, is the platform of the Neem lovers. I am very much honored to get this award from this my beloved organization. Thank you very much, especially Professor Bass, Dr. Nirmala Kothari, their love, passion to Neem, this moving forward, this organization, I hope all of will join. And I know there's a lot of people are here, very big industry people are here. So I will also, all of them, I will request to also bring your hand for the active research on the Neem. So this research, I believe, also help our global Neem industry and propagation of the Neem. Thank you very much.
सर आपको मिलते हैं मिलन मैं किससे बोलूं कौन मिलन मिलन सर ऐसा ना ये वही ऑर्गेनाइज करा रहे हैं प्रॉपर वही देख रहे हैं मिलन सर अगर आपको मिलन सर मैं बता रहा हूँ हाँ नीचे WNO की वो स्टॉल लगी हाँ वहाँ पर जाके आप उमेश सर होंगे और मिलन सर होंगे उनसे एक बार पता करना ये शायद मिला अगर मेरा 3 बैग लास्ट रो में रखा था है ना सर हाँ WNO के स्टॉल पर ये खुद की है ना सेलेक्ट फॉर पोस्टर आपको पोस्टर लगेगा नीचे स्टॉल में पोस्टर हियर हाँ
हमारी इजाजत के बिना यहाँ से हवा भी पास नहीं होनी चाहिए। उन्होंने ऐसी कोई नाकाबंदी पार करके पुलवामा किया था। पुलवामा
Is Neem, the tree of 21st century. Azadirachta indica ajas. It is one of the most suitable and valuable tree species native of tropical and subtropical regions. Today, it has the most potential of any tree of the globe due to its industry. The demand for neem products is growing. Of course, it is a boom for environment too. There is a huge market of neem-based products worldwide. Highest demand is in the pesticide and fertilizer industry. Over the last 30 years, the global price of neem fruits has gone up from 150 to 175 US dollars per ton to the current levels of 400 to 500 US dollars per ton. So we can say that neem industrialization has good potentiality. But what does that mean?
There is a big gap between the supply and the demand. After 2000, the popularity of neem has spread so wide. A day may come when Indians will not have neem for their own use. The amount of demand from abroad and all, it's growing so much. When I started the business, it was not such a popular product. But when days are moving ahead, after 90s, there was a demand for this neem. When Americans registered this product, they wanted to patent this product, and they popularized this product as azadirachtin, which is an organic pesticide ingredient. Growth was very big. Actually, the demand was growing and they were using it. And wherever they used this product, they found it's a wonderful molecule. And in other countries, they could not get it. Though there are other plant products like pyrethrum and all, neem was very different from that. So this product has a lot of popularity and it's still growing. When they started doing this, the other countries like Vietnam, Japan, European countries, and Australia and all, they started using this product, and everybody found this more potent and very helpful product. So that is how I say when they see the market potential and the growth, the inquiries coming year by year and all, I feel that there is a big gap between supply and demand.
Today, almost every other product has neem extract in it, but the supply of these extracts, especially high quality extracts, is limited. To meet the demand of exponentially growing neem market, it is important to spread awareness. The benefits of neem are well known, but it is still not known by many how to obtain high quality and quantity of raw material. This is crucial because the whole chain of supply affects the final product being introduced to the consumer. Good quality final product means more sales, which lead to more demand. This is how any industry grows. Now is the time for Neem. In the recent times, there has been a major shift from chemical to biofertilizer and biopesticide. This has led to an increase in demand of neem oil. Though all the parts of neem tree are beneficial, neem fruits are the most precious as they are not found all year round. To meet commercial demand of neem oil, the neem fruits have to be handled with utmost care because it is the kernel of the neem seed which is filled with our liquid gold. The kernel is the richest source of oil as well as oleochemicals. So, we set out on a journey to find best practices from neem fruit collection to extraction of neem oil and other useful products. Neem fruit is a berry which is green when unripe. As the ripening progresses, the color of the outer skin, that is the pericarp, changes to yellow. On maturity, the fruit is fleshy and filled with sweetish fluid. It is estimated that a fully mature neem tree, which is approximately 8 to 10 years old, will yield 37 to 50 kilograms of fresh fruit, depending on the growing condition. Fresh fruit contains 40 to 50% moisture. So, 40 kilograms of fresh fruit will result in approximately 24 kilograms of dry fruit yield. Neem tree flowers from March to May in different parts of India. As a result, the fruits are mostly available from May to August. The collection of neem fruit in India ranges from meager 1% to maximum 29% of the total theoretical potential of collection. The primary reason for such poor collection are the unorganized plantations and unplanned collection effort. Neem trees are abundantly found in India, but they are scattered in forests, villages, semi-urban, and urban areas. Mostly, neem fruits are collected by tribals and locals who do not have enough knowledge and expertise on how to check and maintain the quality. As a result, the neem fruits collected have to undergo a series of segregation and rejection before reaching neem fruit buyers. There are two major solutions to this problem. Firstly, efforts should be made to educate tribals, farmers, and other locals so that they can be hired as trained neem fruit collectors.
We have to educate the people to collect the raw material in the proper way. The raw material should be free from moisture, stones, sticks, everything. So we have to give the respect to the raw material. We are getting the traders from the North India. They are coming here. We have trained them to how you have to collect the material, how you have to buy the material from the farmers or local collectors or local other small traders like that. So now we are educating. Now it will be improved in future. Yes,
2 or 3 years it will do it.
Secondly, organized neem plantations should be promoted so that standardized instructions for collection can be introduced and implemented. The main reason to implement harvesting practices is to ensure that neem seeds do not come in contact with the ground. This will ensure that the contamination of seeds by aflatoxin does not occur. Aflatoxins are a serious risk to the health of all users of contaminated products. Harvesting can be performed manually or mechanically. Ripened seeds can be removed by hand into a basket to ensure that no seed is wasted. Large clean tarpaulin, cloth, or layered net sheet should be laid out around the base of each tree being harvested so that any seeds which fall in the process are collected on the same.
You are late. There are no fruits now. However, I can demonstrate you how the women in our neighborhood gather fruits. We lay this net, which is at least 2 feet larger than the canopy, to ensure that all the fruits fall onto the net and not on the ground.
However, in some cases, like for manufacture of neem cake, even fruits or seeds collected from the ground can be accepted after proper cleaning and drying. Manual fruit collection becomes impractical for large-scale operations, so it is more economical to harvest mechanically. This can be achieved by using handheld battery-operated hedge trimmers, vertical mowers attached to a tractor, hand-operated battery-powered vacuum which sucks the ripened fruits from the twigs. In all three operations, a tarpaulin should be used under the tree being harvested so that any fruits collected or dropped can be recovered. By adopting this methodology, it can be clearly demonstrated that we in neem industry are promoting the best practices in our harvesting techniques. The collected fruits can be divided in 4 categories for the purpose of handling. Firstly, wet and damaged fruits which are not suitable for oil extraction are separated. Fresh fruits that have about 50% moisture are washed and laid out to dry in the sun. Storage and transportation of undried fruits is not recommended. Semi-dry fruits are the ones with 20 to 15% moisture. At this stage, they are mostly brought to the market. They are further dried to bring down moisture to less than 10% before storing and processing. If fruits contain less than 9% moisture, they are considered dried fruits, and they can be further divided into 2 categories. Clean fruits free from dirt, dust, leaves, twigs, etc. can be taken for dehulling and decortication. Stock containing foreign material has to undergo further segregation. Once the fruits are collected, it is time to clean them. If there is mud sticking to fruits, it is removed by washing with clean water. This is important to prevent fungal infection such as aflatoxin-producing fungus aspergillus. Washed fruits should then be laid out in the sun on an uncontaminated concrete hard stand. Depulping is the process of removing pulp and skin from the fruit. It is a common knowledge that in southern India, fruits are depulped before storing, whereas in northern parts, dried fruits are stored as such. Depulped fruit is preferred for storage as it can be preserved up to 6 months without any significant deterioration in quality. Depulping is done by storing fresh fruits in a water tank for 4 to 5 days, during which the outer pulp gets decomposed and becomes detachable. At this stage, pulp can be removed manually or fruit is taken to a depulper. Detached pulp is washed off with jets of water and the resultant seed is taken for drying. It is important to dry the fruits or seeds before storing. Ripe fruits contain nearly 50% moisture, along with a lot of carbohydrates in the form of pulp. Improper handling and storage can lead to fermentation.
So the main issue is the best quality before it getting rotten, before it gets burnt, before it gets black. So immediately we should procure so that it may not get, if you keep it for a long time, maybe 10 days, 15 days, or if it gets wet, then the neem fruit generates a lot of heat and it easily absorbs the moisture. So once it is dumped, once it, along with the moisture, if it heats up, it will automatically burn, it will become black, or even the smoke will come out. So that is the condition of the neem fruit. It should be handled properly, otherwise you will lose the entire material. Once I went to the mandi and they dumped the neem fruit bags in the godown and they pulled the shutter. So the person took us to sell the material. When we went there, that godown, the shutter was coming out. इतना पूरा हो गया तो मतलब उसका पूरा burnt हो गया, it's expanded and shutter was broken. The entire metal is black and it's all burnt. It will black like a hair color only. So that is the main reason. So we have to buy the raw material in a good quality raw material to get the quality iron. So that's it. Not the process and they get 1000 ppm or 800 ppm. The same material we buy, bring it here, properly dry it, properly stack it with ventilation, everything. So we get the same around 2000 ppm as a direct. So that way, handling of raw material is the most crucial part of the neem industry.
Neem fruit harvesting in India coincides with onset of monsoon. This makes drying and storage of neem fruits and seeds in a well-ventilated environment challenging. In cloudy, rainy weather, use of fans and hot air blowers is suggested.
It is saved from rainwater.
Okay.
And it is coming with a lot of moisture at the time of fresh arrival. Immediately it has to be dried.
Okay.
But it is people's responsibility to save the material in the right manner. In the market, I'm telling, when it is arrived here, we will save like a gold.
Okay.
Even in the open yard, we can dry. It's expensive there. The safety of this is when immediately rain comes. Now it's a monsoon time. To save the material from the monsoon, we are doing that, number one. Apart from that, we are taking this roof will give you more 10 degrees Celsius. This is putting mainly to invite the more heat temperature inside, and it is to maintain the less humidity. That is why. The screen I told you, when more moisture, all the screen will go up. When it is less moisture, we will down the screen. So transport time, it is take minimum 48 hours to 76 hours. So at the time, if they loaded with more moisture, because of this fermentation and humidity, it will become a color like this. That is why we will educate our suppliers. Don't pack the material with high moisture. Try to reduce the moisture before loading the material, which can save the material, not only the outside of color, inside the kernel, quality, oil recovery, everything.
Dried or semi-dried fruits and seeds are packed in broadly weaved jute bags or mesh bags. These are brought to the markets where oil mills or cake manufacturers buy the desired quality and quantity of fruits and seeds from the collectors.
Is a fine quality.
Okay.
This is the green or biscuit color. This is a fine quality. Is the damage?
Okay.
Take it. Kernel damage.
The rate of raw material depends upon multiple factors. We look for heaps with least foreign material so that we have to spend less time and energy. We take fruits in the fist and shake it. The rattling of kernel inside the fruit or seed tells about the moisture content. Low moisture content is desirable for easier storage. We buy fruits at 20% moisture and then we dry them further to 5% moisture before processing.
During storage, frequent agitation of fruits or seeds is advisable. This is crucial to dissipate heat generated due to auto-oxidation, fermentation of pulp, and respiration of seeds. This agitation can be performed manually or mechanically. The frequency of agitation depends on the moisture content in fruits or seeds. Wet material should not be placed in big heaps for more than a day. Prefer small, fresh heaps. They can also be stacked with proper air circulation. Dried fruits or seeds can be stored in bins or bags that allow good ventilation and dissipate heat.
What we have asked this year is the earlier used to bag it in the jute gunny bag, which allows no ventilation. So this year we asked them to bag it in the onion bags with jolly type, plastic bags, which is much way better. So it doesn't get spoiled easily. So it allows some air circulation. So even if after bringing, even if I stack it for a week or so, it doesn't get spoiled.
Apart from moisture control, it is important to maintain ventilation and sanitation in storage areas. As we discussed before, neem fruits are available for a short period of time just before onset of monsoon. For large-scale producers, it becomes impractical to process the whole stock in 4 to 6 months. So, to distribute the workload throughout the year, it is wise to stock the seeds in cold storage after proper drying and cleaning.
To retain the color and the content, everything, the purpose we are putting in the cold storage, that's all.
This ensures minimum deterioration in the quality of kernel. Once dried, the fruits are ready for further cleaning. They can be fed to revolving screens or reels, mechanical or manual sieves to remove foreign material such as stones, sticks, leaves, sand, dirt from the fruits. These machines separate fruit from foreign material on the basis of shape, size, and density. This process has to be repeated multiple times to get neem fruits free from any foreign materials. Electromagnets can be used to separate clamp iron pieces. During the last stages of cleaning, pneumatic system can be used for aspirating light material.
All the dust from that machine entered into that chamber.
It is well established that neem kernel is the richest source of neem oil or desired oleochemicals for industrial use. Crushing the whole seed to obtain oil is not recommended. This puts extra load on the extractor and also results in poor quality of extraction due to impurities carried along with seed coat. Besides, the yield of the produce is less due to absorption by shells or pulp. Therefore, decortication of seed is desirable before oil extraction. It is a common practice to directly feed dried fruits to decorticator, where kernels get separated from pericarp along with seed coat. Decorticator results in a mix of kernels and husk. In case of depulped seeds, the process is easier and faster as just seed coat is to be removed. This mix is then passed through double-deck vibrating screen machines to get rid of husk and remnant impurities. After repeated cleaning, the kernels are ready for oil extraction.
Huh? Ah, oh.
Finally, the kernels are fed into the oil expeller. For small-scale production, rotary expeller or gunny is used. It can be best described as an iron rod in a large bucket which will press the kernels to expel out oil, and the cake will remain in the bucket. For large-scale production, large oil expellers are used where multiple rollers press the kernels inside a tank to expel oil from one end of the machine and cake is expelled from the other end. In industry, there is a widely prevalent practice of producing cold-pressed oil, which is considered superior in quality. It is extracted by circulating cold water between the rollers to keep the temperature low. It is important to clean the oil expellers regularly. This oil is collected in a sedimentation tank. For 3 to 4 days, the oil is shifted between the tanks to allow unwanted debris to settle down, so that pure neem oil can easily be filtered out. The obtained neem oil can be used in agriculture and other industries after appropriate dilution or can be further processed to extract as a directant. After oil extraction, the remnant kernel can be used to make neem cake, neem powder, or neem pellets. The chemical as well as physical composition of neem cake depends on the raw material used. Manufacturers who are more concentrated towards oil production use the residue kernel as cake. Usually, neem fruits which are not fit for making oil are used to make neem cake. This cake has higher oil content. Here, the journey of neem fruit comes to an end as it takes various forms. To summarize, the neem industry can cater to global markets. The research provides us key to unlock full potential of this amazing tree. Though the theoretical documentations give us precise guidelines and high standards for gathering and handling raw material, the ground reality is different. Financial constraints and lack of this theoretical understanding is a barrier to the neem industry's transformation into what it seeks to be. While initiatives are in place to support structured fruit collection, they are insufficient for a tree like neem that spans such a wide geographic area. Neem plantations that are well organized can assist us in focusing our attention so that targeted solutions can be put into action. Is there any reason that state government or central government should take interest in these projects?
Definitely, they have a lot of land bank, unused land. See, one thing I would like to tell you, any waste land can be converted into a neem plantation. So if actually the government are also trying to develop something. In fact, we had an invitation from the Agriculture Department of Tamil Nadu to promote neem all over the place, and we have given some suggestions to them, and they are working on it. Actually, the temple lands, lot of waste temple lands are there. There also we are suggesting, if anybody is there to develop neem plantation and they once they harvest and if they give the neem seeds, there are corporates to enter into a buyback agreement with them and support them in all ways. There is yet a big potential in this field. The youngsters have should come into this field and they should do a lot of things more, according to me.
Besides workshops, large-scale initiative is required. This documentary is just the first step. This is Neem, the tree of 21st century. As a director indica Jas. It is one of the most suitable and valuable tree species native of tropical and subtropical regions. Today, it has been recognized as having the most potential of any tree of the globe due to its evergreen nature and ability to grow in even the most arid and nutrient-deficient soils. Neem has found its glory worldwide due to its diverse range of actions and uses. Agriculture, human health, animal health. In almost every industry, the demand for neem products is growing. Of course, it is a boon for environment too. There is a huge market of neem-based products worldwide. Highest demand is in the pesticide and fertilizer industry. Over the last 30 years, the global price of neem fruits has gone up from 150 to 175 US dollars per ton to the current levels of 400 to 500 US dollars per ton. So we can say that neem industrialization has good potentiality. But what does that mean?
There is a big gap between supply and demand. After 2000, the popularity of neem has spread so wide. A day may come when Indians will not have neem for their own use. The amount of demand from abroad and all, it's growing so much. When I started the business, it was not such a popular product. But when days are moving ahead, after 90s, there was a demand for this neem. When Americans registered this product, they wanted to patent this product, and they popularized this product as a azadirachtin, which is an organic pesticide ingredient. Growth was very big. Actually, the demand was growing and they were using it. And wherever they used this product, they found it's a wonderful molecule. And in other countries, they could not get it. Though there are other plant products like pyrethrum and all, neem was very different from that. So this product has a lot of popularity and it's still growing. When they started doing this, the other countries like Vietnam, Japan, European countries and Australia and all, they started using this product and everybody found this more potent and very helpful product. So that is how I say when they see the market potential and the growth, the inquiries coming year by year and all, I feel that there is a big gap between supply and demand.
Today, almost every other product has neem extract in it, but the supply of these extracts, especially high quality extracts, is limited. To market, it is important to spread awareness. The benefits of neem are well known, but it is still not known by many how to obtain high quality and quantity of raw material. This is crucial because the whole chain of supply affects the final product being introduced to the consumer. Good quality final product means more sales, which lead to more demand. This is how any industry grows. Now is the time for Neem. In the recent times, there has been a major shift from chemical to biofertilizer and biopesticide. This has led to an increase in demand of neem oil. Though all the parts of neem tree are beneficial, neem fruits are the most precious as they are not found all year round. To meet commercial demand of neem oil, the neem fruits have to be handled with utmost care because it is the kernel of the neem seed which is filled with our liquid gold. The kernel is the richest source of oil as well as oleochemicals. So, we set out on a journey to find best practices from neem fruit collection to extraction of neem oil and other useful products. Neem fruit is a berry which is green when unripe. As the ripening progresses, the color of the outer skin, that is the pericarp, changes to yellow. On maturity, the fruit is fleshy and filled with sweetish fluid. It is estimated that a fully mature neem tree, which is approximately 8 to 10 years old, will yield 37 to 50 kilograms of fresh fruit, depending on the growing condition. Fresh fruit contains 40 to 50% moisture. So, 40 kilograms of fresh fruit will result in approximately 24 kilograms of dry fruit yield. Neem tree flowers from March to May in different parts of India. As a result, the fruits are mostly available from May to August. The collection of neem fruit in India ranges from meager 1% to maximum 29% of the total theoretical potential of collection. The primary reason for such poor collection are the unorganized plantations and unplanned collection effort. Neem trees are abundantly found in India, but they are scattered in forests, villages, semi-urban, and urban areas. Mostly, neem fruits are collected by tribals and locals who do not have enough knowledge and expertise on how to check and maintain the quality. As a result, the neem fruits collected have to undergo series of segregation and rejection before reaching neem fruit buyers. There are two major solutions to this problem. Firstly, efforts should be made to educate tribals, farmers, and other locals so that they can be hired as trained neem fruit collectors.
We have to educate the people to collect the raw material the proper way. The raw material should be free from moisture, stones, sticks, everything. So we have to give the respect to the raw material. We are getting the traders from the North India. They are coming here. We have trained them to how you have to collect the material, how you have to buy the material from the farmers or local collectors or local other small traders like that. So now we are educating. Now it will be improved in future years, 2 or 3 years it will improve.
Secondly, organized neem plantations should be promoted so that standardized instructions for collection can be introduced and implemented. The main reason to implement harvesting practices is to ensure that neem seeds do not come in contact with the ground. This will ensure that the contamination of seeds by aflatoxin does not occur. Aflatoxins are a serious risk to the health of all users of contaminated products. Harvesting can be performed manually or mechanically. Ripened seeds can be removed by hand into a basket to ensure that no seed is wasted. Large clean tarpaulin, cloth, or layered net sheet should be laid out around the base of each tree being harvested so that any seeds which fall in the process are collected on the same.
You are late. There are no fruits now. However, I can demonstrate you how the women in our neighborhood gather fruits. We lay this net, which is at least 2 feet larger than the canopy, to ensure that all the fruits fall onto the net and not on the ground.
However, in some cases, like for manufacture of neem cake, even fruits or seeds collected from the ground can be accepted after proper cleaning and drying. Manual fruit collection becomes impractical for large-scale operations, so it is more economical to harvest mechanically. This can be achieved by using handheld battery-operated hedge trimmers, vertical mowers attached to a tractor, hand-operated battery-powered vacuum which sucks the ripened fruits from the twigs. In all three operations, a tarpaulin should be used under the tree being harvested so that any fruits collected or dropped can be recovered. By adopting this methodology, it can be clearly demonstrated that we in neem industry are promoting the best practices in our harvesting techniques. The collected fruits can be divided in 4 categories for the purpose of handling. Firstly, wet and damaged fruits which are not suitable for oil extraction are separated. Fresh fruits that have about 50% moisture are washed and laid out to dry in the sun. Storage and transportation of undried fruits is not recommended. Semi-dry fruits are the ones with 20 to 15% moisture. At this stage, they are mostly brought to the market. They are further dried to bring down moisture to less than 10% before storing and processing. If fruits contain less than 9% moisture, they are considered dried fruits, and they can be further divided into 2 categories. Clean fruits free from dirt, dust, leaves, twigs, etc. can be taken for dehulling and decortication. Stock containing foreign material has to undergo further segregation. Once the fruits are collected, it is time to clean them. If there is mud sticking to fruits, it is removed by washing with clean water. This is important to prevent fungal infection such as aflatoxin-producing fungus aspergillus. Washed fruits should then be laid out in the sun on an uncontaminated concrete hard stand. Depulping is the process of removing pulp and skin from the fruit. It is a common knowledge that in southern India, fruits are depulped before storing, whereas in northern parts, dried fruits are stored as such. Depulped fruit is preferred for storage as it can be preserved up to 6 months without any significant deterioration in quality. Depulping is done by storing fresh fruits in a water tank for 4 to 5 days, during which the outer pulp gets decomposed and becomes detachable. At this stage, pulp can be removed manually or fruit is taken to a depulper. Detached pulp is washed off with jets of water and the resultant seed is taken for drying. It is important to dry the fruits or seeds before storing. Ripe fruits contain nearly 50% moisture, along with a lot of carbohydrates in the form of pulp. Improper handling and storage can lead to fermentation.
So the main issue is the best quality before it getting rotten, before it gets burnt, before it gets black. So immediately we should procure so that it may not get, if you keep it for a long time, maybe 10 days, 15 days, or if it gets wet, then the neem fruit generates a lot of heat and it easily absorbs the moisture. So once it is dumped, along with the moisture, if it heats up, it will automatically burn, it will become black, or even the smoke will come out. So that is the condition of the neem fruit. It should be handled properly, otherwise you will lose the entire material. Once I went to the mandi and they dumped the neem fruit bags in the godown and they pulled the shutter. So the person took us to sell the material. When we went there, that godown, the shutter was coming out. इतना पूरा हो गया तो मतलब उसका पूरा burnt हो गया, expanded and shutter was broken. The entire metal is black and it's all burnt. It will black like a hair color only. So that is the main reason. So we have to buy the raw material in a good quality raw material to get the quality iron. So that's it. Not the process and they get 1000 ppm or 800 ppm. The same material we buy, bring it here, properly dry it, properly stack it with ventilation, everything. So we get the same around 2000 ppm as a direct. So that way, handling of raw material is most crucial part of the neem industry.
Neem fruit harvesting in India coincides with onset of monsoon. This makes drying and storage of neem fruits and seeds in a well-ventilated environment challenging. In cloudy, rainy weather, use of fans and hot air blowers is suggested.
It is saved from rainwater.
Okay.
And it is coming with a lot of moisture at the time of fresh arrival. Immediately it has to be dried.
Okay.
But it is people's responsibility to save the material in the right manner. In the market, I'm telling, when it is arrived here, we will save like a gold.
Okay.
Even in the open yard, we can dry. It's expensive here. The safety of this is when immediately rain comes. Now it's a monsoon time. To save the material from the monsoon, we are doing that, number one. Apart from that, we are taking this roof will give you more 10 degrees Celsius. This is putting mainly to invite the more heat temperature inside, and it is to maintain the less humidity. That is why. The screen I told you, na, when more moisture, all the screen will go up. When it is less moisture, we will down the screen. The transport time, it is take minimum 48 hours to 76 hours. So at the time, if they loaded with more moisture, because of this fermentation, and humidity, it will become a color like this. That is why we will educate our suppliers. Don't pack the material with a high moisture. Try to reduce the moisture before loading the material, which can save the material, not only the outside of color, inside the kernel, quality, oil recovery, everything.
Dried or semi-dried fruits and seeds are packed in broadly weaved jute bags or mesh bags. These are brought to the markets where oil mills or cake manufacturers buy the desired quality and quantity of fruits and seeds from the collectors.
Here's the fine quality.
Okay.
This is the green or biscuit color. This is the fine quality. Is the damage.
Okay.
Take it. Kernel damage.
The rate of raw material depends upon multiple factors. We look for heaps with least foreign material so that we have to spend less time and energy. We take fruit silver fist and shake it. The rattling of kernel inside the fruit or seed tells about the moisture content. Low moisture content is desirable for easier storage. We buy fruits at 20% moisture and then we dry them further to 5% moisture before processing.
During storage, frequent agitation of fruits or seeds is advisable. This is crucial to dissipate heat generated due to auto-oxidation, fermentation of pulp, and respiration of seeds. This agitation can be performed manually or mechanically. The frequency of agitation depends on the moisture content in fruits or seeds. Wet material should not be placed in big heaps for more than a day. Prefer small, fresh heaps. They can also be stacked with proper air circulation. Dried fruits or seeds can be stored in bins or bags that allow good ventilation and dissipate heat.
What we have asked this year is the earlier used to bag it in the jute gunny bag, which allows no ventilation. So this year we asked them to bag it in the onion bags with jolly type, plastic bags, which is much way better. So it doesn't get spoiled easily. So it allows some air circulation. So even if after bringing, even if I stack it for a week or so, it doesn't get spoiled.
Apart from moisture control, it is important to maintain ventilation and sanitation in storage areas. As we discussed before, neem fruits are available for a short period of time just before onset of monsoon. For large-scale producers, it becomes impractical to process the whole stock in 4 to 6 months. So, to distribute the workload throughout the year, it is wise to stock the seeds in cold storage after proper drying and cleaning.
To retain the color and the content, everything, the purpose we are putting in the cold soil, that's all.
This ensures minimum deterioration in the quality of kernel. Once dried, the fruits are ready for further cleaning. They can be fed to revolving screens or reels, mechanical or manual sieves to remove foreign material such as stones, sticks, leaves, sand, dirt from the fruits. These machines separate fruit from foreign material on the basis of shape, size, and density. This process has to be repeated multiple times to get neem fruits free from any foreign materials. Electromagnets can be used to separate clamp iron pieces. During the last stages of cleaning, pneumatic system can be used for aspirating light material.
All the dust from that machine entered into that chamber.
It is well established that neem kernel is the richest source of neem oil or desired oleochemicals for industrial use. Crushing the whole seed to obtain oil is not recommended. This puts extra load on the extractor and also results in poor quality of extraction due to impurities carried along with seed coat. Besides, the yield of the produce is less due to absorption by shells or pulp. Therefore, decortication of seed is desirable before oil extraction. It is a common practice to directly feed dried fruits to decorticator, where kernels get separated from pericarp along with seed coat. Decorticator results in a mix of kernels and husk. In case of depulped seeds, the process is easier and faster as just seed coat is to be removed. This mix is then passed through double-deck vibrating screen machines to get rid of husk and remnant impurities. After repeated cleaning, the kernels are ready for oil extraction. Finally, the kernels are fed into the oil expeller. For small-scale production, rotary expeller or gunny is used. It can be best described as an iron rod in a large bucket which will press the kernels to expel out oil, and the cake will remain in the bucket. For large-scale production, large oil expellers are used where multiple rollers press the kernels inside a tank to expel oil from one end of the machine and cake is expelled from the other end. In industry, there is a widely prevalent practice of producing cold-pressed oil, which is considered superior in quality. It is extracted by circulating cold water between the rollers to keep the temperature low. It is important to clean the oil expellers regularly. This oil is collected in a sedimentation tank. For 3 to 4 days, the oil is shifted between the tanks to allow unwanted debris to settle down, so that pure neem oil can easily be filtered out. The obtained neem oil can be used in agriculture and other industries after appropriate dilution or can be further processed to extract as a directin. After oil extraction, the remnant kernel can be used to make neem cake, neem powder, or neem pellets. The chemical as well as physical composition of neem cake depends on the raw material used. Manufacturers who are more concentrated towards oil production use the residue kernel as cake. Usually, neem fruits which are not fit for making oil are used to make neem cake. This cake has higher oil content. Here, the journey of neem fruit comes to an end as it takes various forms. To summarize, the neem industry can cater to global markets. The research provides us key to unlock full potential of this amazing tree. Though the theoretical documentations give us precise guidelines and high standards for gathering and handling raw material, the ground reality is different. Financial constraints and lack of this theoretical understanding is a barrier to the neem industry's transformation into what it seeks to be. While initiatives are in place to support structured fruit collection, they are insufficient for a tree like neem that spans such a wide geographic area. Neem plantations that are well organized can assist us in focusing our attention so that targeted solutions can be put into action.
Is there any reason that state government or central government should take interest in these projects?
Definitely, they have a lot of land bank, unused land. See, one thing I would like to tell you, any waste land can be converted into a neem plantation. So if actually the government are also trying to develop something. In fact, we had an invitation from the Agriculture Department of Tamil Nadu to promote neem all over the place, and we have given some suggestions to them, and they are working on it. Actually, the temple lands, lot of waste temple lands are there. There also we are suggesting, if anybody is there to develop neem plantation, and once they harvest and if they give the neem seeds, there are corporates to enter into a buyback agreement with them and support them in all ways. There is yet a big potential in this field. The youngsters should come into this field, and they should do a lot of things more, according to me.
Besides workshops, large-scale initiative is required. This documentary is just the first step. This is Neem, the tree of 21st century. As a director indica Jas. It is one of the most suitable and valuable tree species native of tropical and subtropical regions. Today, it has been recognized as having the most potential of any tree of the globe due to its evergreen nature and ability to grow in even the most arid and nutrient deficient soils. Neem has found its glory worldwide due to its diverse range of actions and uses. Agriculture, human health, animal health. In almost every industry, the demand for neem products is growing. Of course, it is a boon for environment too. There is a huge market of neem-based products worldwide. Highest demand is in the pesticide and fertilizer industry. Over the last 30 years, the global price of neem fruits has gone up from 150 to 175 US dollars per ton to the current levels of 400 to 500 US dollars per ton. So we can say that neem industrialization has good potentiality. But what does that mean?
There is a big gap between supply and demand. After 2000, the popularity of Neem has spread so wide. A day may come when Indians will not have Neem for their own use. The amount of demand from abroad and all, it's growing so much. When I started the business, it was not such a popular product. But when days are moving ahead, after 90s, there was a demand for this Neem. When Americans registered this product, they wanted to patent this product, and they popularized this product as a azadirachtin, which is an organic pesticide ingredient. Growth was very big. Actually, the demand was growing and they were using it. And wherever they used this product, they found it's a wonderful molecule. And in other countries, they could not get it. Though there are other plant products like pyrethrum and all, Neem was very different from that. So this product has a lot of popularity and it's still growing. When they started doing this, the other countries like Vietnam, Japan, European countries and Australia and all, they started using this product, and everybody found this more potent and very helpful product. So that is how I say, when they see the market potential and the growth, the inquiries coming year by year and all, I feel that there is a big gap between supply and demand.
Today, almost every other product has neem extract in it, but the supply of these extracts, especially high quality extracts, is limited. To meet the demand of exponentially growing neem market, it is important to spread awareness. The benefits of neem are well known, but it is still not known by many how to obtain high quality and quantity of raw material. This is crucial because the whole chain of supply affects the final product being introduced to the consumer. Good quality final product means more sales, which lead to more demand. This is how any industry grows. Now is the time for Neem. In the recent times, there has been a major shift from chemical to biofertilizer and biopesticide. This has led to an increase in demand of neem oil. Though all the parts of neem tree are beneficial, neem fruits are the most precious, as they are not found all year round. To meet commercial demand of neem oil, the neem fruits have to be handled with utmost care, because it is the kernel of the neem seed which is filled with our liquid gold. The kernel is the richest source of oil as well as oleochemicals. So, we set out on a journey to find best practices from neem fruit collection to extraction of neem oil and other useful products. Neem fruit is a berry which is green when unripe. As the ripening progresses, the color of the outer skin, that is the pericarp, changes to yellow. On maturity, the fruit is fleshy and filled with sweetish fluid. It is estimated that a fully mature neem tree, which is approximately 8 to 10 years old, will yield 37 to 50 kilograms of fresh fruit, depending on the growing condition. Fresh fruit contains 40 to 50% moisture. So, 40 kilograms of fresh fruit will result in approximately 24 kilograms of dry fruit yield. Neem tree flowers from March to May in different parts of India. As a result, the fruits are mostly available from May to August. The collection of neem fruit in India ranges from meager 1% to maximum 29% of the total theoretical potential of collection. The primary reason for such poor collection are the unorganized plantations and unplanned collection effort. Neem trees are abundantly found in India, but they are scattered in forests, villages, semi-urban, and urban areas. Mostly, neem fruits are collected by tribals and locals who do not have enough knowledge and expertise on how to check and maintain the quality. As a result, the neem fruits collected have to undergo series of segregation and rejection before reaching neem fruit buyers. There are two major solutions to this problem. Firstly, efforts should be made to educate tribals, farmers, and other locals so that they can be hired as trained neem fruit collectors.
We have to educate the people to collect the raw material the proper way. The raw material should be free from moisture, stones, sticks, everything. So we have to give the respect to the raw material. We are getting the traders from the North India. They are coming here. We have trained them to how you have to collect the material, how you have to buy the material from the farmers or local collectors or local other small traders like that. So now we are educating. Now it will be improved in future years, 2 or 3 years it will improve.
Secondly, organized neem plantations should be promoted so that standardized instructions for collection can be introduced and implemented. The main reason to implement harvesting practices is to ensure that neem seeds do not come in contact with the ground. This will ensure that the contamination of seeds by aflatoxin does not occur. Aflatoxins are a serious risk to the health of all users of contaminated products. Harvesting can be performed manually or mechanically. Ripened seeds can be removed by hand into a basket to ensure that no seed is wasted. Large clean tarpaulin, cloth, or layered net sheet should be laid out around the base of each tree being harvested so that any seeds which fall in the process are collected on the same.
You are late. There are no fruits now. However, I can demonstrate you how the women in our neighborhood gather fruits. We lay this net, which is at least 2 feet larger than the canopy, to ensure that all the fruits fall onto the net and not on the ground.
However, in some cases, like for manufacture of neem cake, even fruits or seeds collected from the ground can be accepted after proper cleaning and drying. Manual fruit collection becomes impractical for large-scale operations, so it is more economical to harvest mechanically. This can be achieved by using handheld battery-operated hedge trimmers, vertical mowers attached to a tractor, hand-operated battery-powered vacuum which sucks the ripened fruits from the twigs. In all three operations, a tarpaulin should be used under the tree being harvested so that any fruits collected or dropped can be recovered. By adopting this methodology, it can be clearly demonstrated that we in Neem industry are promoting the best practices in our harvesting techniques. The collected fruits can be divided in 4 categories for the purpose of handling. Firstly, wet and damaged fruits which are not suitable for oil extraction are separated. Fresh fruits that have about 50% moisture are washed and laid out to dry in the sun. Storage and transportation of undried fruits is not recommended. Semi-dry fruits are the ones with 20 to 15% moisture. At this stage, they are mostly brought to the market. They are further dried to bring down moisture to less than 10% before storing and processing. If fruits contain less than 9% moisture, they are considered dried fruits, and they can be further divided into 2 categories. Clean fruits free from dirt, dust, leaves, twigs, etc. can be taken for dehulling and decortication. Stock containing foreign material has to undergo further segregation. Once the fruits are collected, it is time to clean them. If there is mud sticking to fruits, it is removed by washing with clean water. This is important to prevent fungal infection such as aflatoxin-producing fungus Aspergillus. Washed fruits should then be laid out in the sun on an uncontaminated concrete hard stand. Depulping is the process of removing pulp and skin from the fruit. It is a common knowledge that in southern India, fruits are depulped before storing, whereas in northern parts, dried fruits are stored as such. Depulped fruit is preferred for storage as it can be preserved up to 6 months without any significant deterioration in quality. Depulping is done by storing fresh fruits in a water tank for 4 to 5 days, during which the outer pulp gets decomposed and becomes detachable. At this stage, pulp can be removed manually or fruit is taken to a depulper. Detached pulp is washed off with jets of water and the resultant seed is taken for drying. It is important to dry the fruits or seeds before storing. Ripe fruits contain nearly 50% moisture, along with a lot of carbohydrates in the form of pulp. Improper handling and storage can lead to fermentation.
So the main issue is the best quality before it getting rotten, before it gets burnt, before it gets black. So immediately we should procure so that it may not get, if you keep it for a long time, maybe 10 days, 15 days, or if it gets wet, then because the neem fruit generates a lot of heat and it easily absorbs the moisture. So once it is dumped, once it, along with the moisture, if it heats up, it will automatically burn, it will become black, or even the smoke will come out. So that is the condition of the neem fruit. It should be handled properly. Otherwise, you will lose the entire material. Once I went to the mandi and they dumped the neem fruit bags in the godown and they pulled the shutter. So the person took us to sell the material. When we went there, that godown, the shutter was coming out. इतना पूरा हो गया तो मतलब उसका पूरा burnt हो गया, it's expanded and shutter was broken. The entire metal is black and it's all burnt. It will black like a hair color only. So that is the main reason. So we have to deal in a good quality raw material to get the quality iron. So that's it. Not the process and they get 1000 ppm or 800 ppm. The same metal we bring it here, properly dry it, properly stack it with ventilation, everything. So we get the same around 2000 ppm as a direct. So that way, handling of raw material is most crucial part in the neem industry.
Neem fruit harvesting in India coincides with onset of monsoon. This makes drying and storage of neem fruits and seeds in a well-ventilated environment challenging. In cloudy, rainy weather, use of fans and hot air blowers is suggested.
It is saved from rainwater.
Okay.
And it is coming with a lot of moisture at the time of fresh arrival. Immediately it has to be dried.
Okay.
But it is people's responsibility to save the metal in the right manner. In the market, I'm telling, when it is arrived here, we will save like a gold. Even in the open yard, we can dry. It's expensive there. The safety of this is when immediately rain comes. Now it's a monsoon time. To save the material from the monsoon, we are doing that, number one. Apart from that, we are taking this roof will give you more 10 degrees Celsius. This is putting mainly to invite the more heat temperature inside, and it is to maintain the less humidity. That is why. The screen I told you, na, when more moisture, all the screen will go up. When it is less moisture, we will down the screen. The transport time, it is take minimum 48 hours to 76 hours. So at the time, if they loaded with more moisture, because of this fermentation and humidity, it will become a color like this. That is why we will educate our suppliers. Don't pack the material with high moisture. Try to reduce the moisture before loading the material, which can save the material, not only the outside of color, inside the kernel, quality, oil recovery, everything.
Dried or semi-dried fruits and seeds are packed in broadly weaved jute bags or mesh bags. These are brought to the markets where oil mills or cake manufacturers buy the desired quality and quantity of fruits and seeds from the collectors.
Is the fine quality.
Okay.
This is the green or biscuit color. This is the fine quality. So is the damage.
Okay.
Take it. Kernel damage.
The rate of raw material depends upon multiple factors. We look for heaps with least foreign material so that we have to spend less time and energy. We take fruit silver fist and shake it. The rattling of kernel inside the fruit or seed tells about the moisture content. Low moisture content is desirable for easier storage. We buy fruits at 20% moisture and then we dry them further to 5% moisture before processing.
During storage, frequent agitation of fruits or seeds is advisable. This is crucial to dissipate heat generated due to auto-oxidation, fermentation of pulp, and respiration of seeds. This agitation can be performed manually or mechanically. The frequency of agitation depends on the moisture content in fruits or seeds. Wet material should not be placed in big heaps for more than a day. Prefer small, fresh heaps. They can also be stacked with proper air circulation. Dried fruits or seeds can be stored in bins or bags that allow good ventilation and dissipate heat.
What we have asked this year is the earlier used to bag it in the jute gunny bag, which allows no ventilation. So this year we asked them to bag it in the onion bags with jolly type, plastic bags, which is much way better. So it doesn't get spoiled easily. So it allows some air circulation. So even if after bringing, even if I stack it for a week or so, it doesn't get spoiled.
Apart from moisture control, it is important to maintain ventilation and sanitation in storage areas. As we discussed before, neem fruits are available for a short period of time just before onset of monsoon. For large-scale producers, it becomes impractical to process the whole stock in 4 to 6 months. So, to distribute the workload throughout the year, it is wise to stock the seeds in cold storage after proper drying and cleaning.
To retain the color and the content, everything, the purpose we are putting in the cold storage, that's all.
This ensures minimum deterioration in the quality of kernel. Once dried, the fruits are ready for further cleaning. They can be fed to revolving screens or reels, mechanical or manual sieves to remove foreign material such as stones, sticks, leaves, sand, dirt from the fruits. These machines separate fruit from foreign material on the basis of shape, size, and density. This process has to be repeated multiple times to get neem fruits free from
Good evening. Good afternoon, ladies and gentlemen. Our chief guest for the day has arrived, and along with him, Dr. Nisha, Dr. Arpi Singh, and Dr. Akhilesh Gupta, and others are also on the dais. Now I request our Dr. Kothari and Dr. Vyas to welcome the chief guest by offering a bouquet and shawl and memento. Now I request Dr. Venkatesh to welcome Dr. Barik. No, he's not there. Okay, sorry. I request Dr. Venkatesh to welcome Dr. Nisha. Yeah, now I request Mr. Girish Soman. He's not there. Yeah, okay. I request Dr. Venkatesh to do the honors to Dr. R.P. Singh. And I request Mr. Vyas to do the honors to Dr. Akhilesh Gupta. Thank you.
Respected chief guest of the program, Dr. S.K. Chaudhary Sahab, DDG NRM ICAR, Dr. Akhilesh Gupta ji, Professor Parikh, Dr. Nisha Mehndiratta ji, Professor R.B. Singh ji, other dignitaries, delegates, ladies and gentlemen, I extend a very warm welcome to you all, sir and madam. After listening to the wonderful presentation yesterday, we are here for the valedictory function and we look forward to the golden words from all of you, sir, which would certainly help us to prepare roadmap for our future action plans. With these very small things, I take my seat. I thank you very much. Jai Hind, Jai Bharat.
Respected dignitaries on dais and our respected delegate speakers, I have privilege to present the report on second Neem Summit 2024. The summit was inaugurated by Dr. Himanshu Pathak ji, DG ICAR, along with guest of honor Dr. P.K. Singh, Agricultural Commissioner, Government of India, and Justice K.K. Tater, Chairman, Human Rights Commission, Government of Maharashtra. Post-inauguration, the keynote address was delivered by Mr. Klaus Forlow from Canada, who highlighted the importance of Neem, especially in human health. Subsequently to that, presentation by 11 invited speakers and 13 presentations by contributed papers. Topics of all the presentation covered all the important aspects of Neem and its use for the commercial application. Out of these presentations, 4 presentations were online from India, from Germany, from Tanzania, and from Spain. This was the first time that we used hybrid platform for this summit. In addition to the scientific presentations, 8 awards were also given to award for making significant contribution to the promote the Neem philosophy among the common population. I thank you all. I thank you all, our guests, for giving us opportunity to show what we have and what Neem is. And really, I thank you all our sponsors, everyone who are involved in making this event a successful event. Thank you.
Now we invite our guest of honor, Dr. Nisha Mandiratta, Executive Director of the Indo-US Science and Technology Forum, located in New Delhi. So, ma'am, please.
Very good afternoon to everyone. Respected dignitaries on the dais, off dais. It's real my pleasure to be here with all of you on this very important event. When Dr. Arunachalam asked me to be here, so first of all, I was really amazed to see the Neem Summit and Global Neem Trade Fair, because being a student of IRI for so long, and today really like I'm getting little nostalgic as well, because when I met Dr. Chaudhary, DDG, ICAR, NRM, and while talking, we came to know that we were more or less together at the same batch. So that itself is so heartening to me. Dr. Balaji is sitting next to me. My friend Nutan, I could see here. So many memories are here with the IRI. And I remember as a student also many workshops, many summits we have attended here, especially the Kisan Mela, I cannot forget every year which happens here. But this Neem Summit again, I was not aware of it. And when I asked Dr. Arunachalam, please do let me know before coming to this event, that what is it exactly. So I am really so happy to see about WNO and the kind of organization which is there for Neem, to not only to look into, I think, its conservation and management, but also to take this breed forward, no, in many ways. And this organization of this fair, this summit, I think is one step towards that only, that it is creating not only the awareness among many people. All of us know, being Indian, the importance of neem. It is one of our herbal medicine as well, which is now getting very, very popular in urban areas, having its immense value, medical values and other values as well. Even for environment, it's so environment friendly. It gives us so much of good breeze and looks into the hazard caused by pollution and climate. But I will not talk so much about it right now, because all of you have already deliberated on it. And as already in the summary report, which was shared by Secretary General WNO, we came to know that many talks, many sessions were held with regard to the importance of Neem. And I am very happy to see the kind of work which is being taken up by CAFRI, Central Agroforestry Research Institute, Jhansi. Not only with Neem, but many activities are there in which this organization is doing wonderful work. And when I asked them to just tell me a little bit, so it was very heartening to see that they have already, like, they are serving as a national field gene bank of Neem germoplasm, for in which many assassin and gene bank for like which have been taken from many states, Maharashtra, Andhra Pradesh, Rajasthan, and the list is very long, which I think all of you are aware of it. But for me also, it is one of the very focused and good news that CAFRI is working so much in the germplasm of neem. And also now many products of neem are also being promoted by them. And I feel that CAFRI has been doing wonderful work, because my association with them, especially Dr. Arunachalam, is from this NRM time. 10 to 12 years back, when we started our national mission on climate change, there was one mission on agriculture. So that was taken care by him as mission director. And at that time, me and Dr. Akhilesh Gupta, who is senior advisor there in DST and secretary SCRB, So I had a fortune to work with him to implement this particular mission. So at that time, many things with regard to agriculture came across, and rather we have built up a program which was on climate change impact on agriculture at that point of a time. And the workshop was held here only in the same campus. So that's why I cannot forget. And many good projects and outcomes were there. And I feel that our that work only is being recognized by the society here. And I also took this opportunity to thank the Society for Science of Climate Change and Sustainable Environment for recognizing our work and honoring us with the recognition in climate change and environmental sustainability. I feel now we have this responsibility to take this cause further. And recently, I have joined Indo-US Forum as Executive Director, and I really feel that these activities I will be very happy to take forward together, whatever way it is possible. Thank you so much.
Thank you, Madam, for those words of wisdom, and then recalling the golden periods of yours and the previous assignment that we all cherished working with you. Thank you so much for those words of wisdom. Now, we request Professor R.P. Singh, the former chairman of Uttar Pradesh State Environment Impact Assessment Authority and a retired professor from Central University of Lucknow. He is also the chairperson of the Society for Science of Climate Change and Sustainable Environment, New Delhi. So Professor R.P. Singh is before us for his words of wisdom.
Thank you, Professor Arunachalam. Good afternoon, honorable dignitaries on the dais, chief guest, Dr. S.K. Chaudhary ji, Dr. Akhilesh Gupta ji, Dr. Mehdi Raza ji, our colleague on the dais who are associated with this Neem conference, and dignitaries of the dais, friends, awardees. It is privilege for us to be here and to invite you who are awardees for the Society for Science of Climate Change and Environmental Sustainability. The society, which was registered in Delhi long back in 2009, is publishing a journal also, and Journal of Climate Change and Environmental Sustainability, and is doing a lot of exploration and work in the field of climate change and environmental sustainability. Friends, we are grateful to the organizers of World Neem Conference, this Neem Summit and Global Neem Trade Fair, who have included this award ceremony of this society in its valedictory session. The benefit of science to society has to be maximized. I feel that the challenges are still ahead. We have been just watching a video here where we could see that the production of any biological product, then maintenance, its transport, then its conversion into product, and then its marketing, and then after, rather through all the process, whatever waste is generated, getting utilization of that waste in a manner where minimum emissions and minimum pollution will occur and the product can be beneficial, so that we can develop the green circular economy, which is a nature-based solution for the future, is very, very challenging. Scientists are required to give their input in collaboration to all other stakeholders to materialize this whole concept in a real sense.
I feel that we are uh, facing a lot of challenges to change the paradigm of developmental patterns so that we can reduce climate change and we can utilize the natural resources our country have. So with these words, I am grateful to you all who are organizers of this mega event and who are here associated with the Climate Change Society and the Neem Foundation. And I appeal you all to work for the better future of the Mother Earth so that we can survive. Otherwise, the way we are going, we may not be in comfortable position in recent future. Thank you.
Thank you, Professor R.P. Singh, for those guiding words that you placed before all of us on the minimization and reduction of climate change impacts, and also Neem as a nature-based solution for all climate amelioration. Thank you so much for that. Friends, we have now with us one of the eminent personalities and a guiding force for development of science and technology and innovation in this country, Dr. Akhilesh Gupta. He has been important key person for all policy processes of the Ministry of Science and Technology. And he is here with us. It's a great pleasure for all of us. Thank you so much for accepting our invitation, sir. So the floor is yours.
Chief guest of today's event, Dr. S.K. Chaudhary, DDG ICR, Professor R.K. Singh, Dr. Arunachalam, Dr. Nisha Mandiratta, all other distinguished dignitaries on the dais, the constituents, ladies and gentlemen. A very good afternoon to all of you. Let me thank the organizer for having invited me for this validity session. The very important conference that you had organized by the World Neem Organization and the ICR Central Agroforestry Research Institute, Jhansi. So my in fact compliment to the organizer for organizing such an important event, and I'm sure the constituent must have been greatly benefited by the deliberations. We know the importance of neem, and not just as medicinal plant, but also in the range of different areas like agriculture, the forestry, medicine, and industry, all put together in wider application of neem. And India has been proud to be having a variety of neems for the various purposes that we do. I'm very happy to be to have been part of your organizing the facilitation for Society for Science of Climate Change and Sustainable Agriculture, Sustainable Environment. Climate change is in fact going to be very, very, I would say, impactful in time to come. In fact, the latest report from IPCC clearly mentioned that India is going to be very severely impacted by climate change in all its walks of life. The three areas that are going to be significant so far as India is concerned is agriculture, water, and health. And therefore, because it is the very India's lifeline is agriculture, and if it is impacted by events like climate change, it will impact the food security and in the water security in terms of water and also the health of human being as well as the health of everything else, all of the species. So that being the case, we need to be doing something very serious in this issue. And the society like an SSCE can contribute in a big way in making people aware of the consequence of climate change. In fact, one of the slogan and dictum that our Prime Minister had given that at the UNFCCC that we must have a different lifestyle. Climate change is largely driven by lifestyle. India has been quite sustainable in terms of its lifestyle. This is determined through various measurements that India has been kind of quite sync with the environment. And I'm not going to detail because of time, but India can become the torch bearer to the world and become the leader to make entire world following in this area of better lifestyle. I think once again I thank the organizer for having invited me for this very important event. I wish this conference as well as the SSCE all the very best for the future. Thank you.
Thank you so much, sir, for those guiding words. And you have always been a guiding force for all of us in this sphere of climate change research, which is being constructively, inclusively coordinated through the Department of Science and Technology. Thank you so much for being with us and gracing the occasion, sir. Now the organizers, the World Neem Organization and the ICAR Central Agroforestry Research Institute has unanimously agreed to get some feedbacks from the delegates, from the companies and the technical participants. So we can take 2 feedbacks voluntarily. Go ahead. So interested feedback givers may come to the stage. Anybody? Yeah, please come. Yeah, please come. He's interested. उसके बाद ठीक है. Please.
Excellencies, my name is Martin Voller-Wühlbecke. I'm here from Paraguay in South America. While I'm with my company member of the World Neem Organization, I think it is appropriate that a non-Indian speaks some warm words to this Indian group. I'm most impressed and by the hearty welcome that you extend to all of the worldwide Neem community here in Delhi and in India. It has been a fantastically well-organized conference in a generous setting. So my deep respect to the Indian authorities that enable such a research institute. And I want to especially thank these people from the World Neem Organization, Dr. Vias, Dr. Kotari, and all the other people that dedicate so much of their time and energy to the good cause. Personally, I can only say I've learned so much, and I think I have made some very valuable friendships and business contacts here. So I hope this event can be repeated. If it would be in Delhi in this very setting in a few years' time, I would come if I would be invited again. Thank you very much.
Thank you very much for that wonderful feedback, and we will try to scale up the activity as we go along in the coming years. Thank you. Dr. Yadav, R.P.S. Yadav from IFCO.
Good afternoon, everyone. Respected guests, actually, when I discussed with this Neem seminar with Dr. Vyasa, it was little bit not comfortable because we have heard a lot of seminars on different issues, but this is a different kind of seminar which I saw here, and I also impressed to see the work of different scientists, different industrialists, and one of the presentation yesterday was the initiative took by these SHG members in Madhya Pradesh to take plantations as well as the collection of neem seed. So I think this is required, and a lot of work has been promoted or doing by the different agencies. So I learned a lot of things, and I hope, and I also thank Dr. Vyas to organize this kind of seminar, and we were happy to share our thoughts also because it was open, and when the presenters were made the presentation during this question hours, lot of people have raised their issues, and I think this is a good environment, and hopefully this Neem seminar will give a path for the betterment of environment. Thank you so much.
Thank you so much for those feedback. We take it positively and constructively. We will grow together. Now I have something to share. Dr. Dwivedi, just 1 minute. Now I get back to the felicitation part and the award ceremony that we plan to do during the valedictory session of this Neem Summit. As rightly put by Professor R.P. Singh, the President of the Society for Science of Climate Change and Sustainable Environment, that we wholeheartedly thank the WNO for providing a space in this session to also accommodate the conferment of the awards that we regularly give. And let the Neem be the witness for all of this. That's what we proudly feel. And you would see the entire decoration is Neemi in nature. Even your flower bouquets, yes, it's not flower bouquet, it is a Neemi flower bouquet. so Neemification has happened all over. So thanks to Neem itself. And with this, I would call upon Dr. R.P. Dwivedi to who will be announcing the category of awards and the name of the awardee, who will come and receive the awards from our chief guests, guest of honors, and all the dais keepers. So I request all the dais keepers to please come to the front of the dais. So Dr. Dwivedi.
Thank you, sir. Namaskar and good afternoon to all. I would like to request the dignitaries on dais. So please kindly give the awards of the Society for Science of Climate Change and Sustainable Environment, SSCE. So the first, starting is award for outstanding contribution to environmental leadership and social responsibility. This individual category, I would like to request this award goes to Dr. Yogesh Gokhale, TERI University, New Delhi. Please come and receive the awards from the dignitaries. Dr. Gokhale, please. Next award is Award for Outstanding Contribution to Environmental Leadership and Social Responsibility under institutional category, and this goes to We Care Foundation, Guwahati, Assam. So representative from We Care Foundation, please come and receive the award. Now, the next category is woman scientist above 45. Awardees are requested to come by left hand, right hand side and go by this left. So first one is Dr. Monalisa Sen, ICLEI, New Delhi. Madam, come from that side. And the second Young Scientist Award for women, Professor Poonam, retired professor, CSKHPKV, Palampur. Then next series, women scientist below 45. The first awardee is Dr. Pooja Gokhale Sinha, Venkateswara College, Delhi. And the second women scientist awardee is Dr. M. Kiranmai Reddy, GITAM University, Vizag. बताइए मैडम, आर्टिकल की तरह। Then Young Scientist Awards, Dr. Vinay Kumar Gadam, VR Siddhartha Engineering College, Vijayawada. Young Scientist Award category. Then next on the series, Dr. Hridayesh Ragi, scientist from ICAR-CAFRI, Jhansi. Then Dr. Rinku Singh, BHU, Varanasi. Then next award for outstanding contribution in climate science and environment sustainability, Dr. Nisha Mendiratta, Executive Director, IUSSTF, New Delhi. Then foreign fellows are there with us. Dr. R.B. Sinha, Senior Policy Advisor, FAO, India Office, New Delhi. Then next, Prakash Kumar Jha ji, Mississippi State University, USA. Now, the society gives fellowship award also. Next, Dr. Akhilesh Gupta, Senior Advisor, DST, New Delhi. So please come and receive the award. Dr. Akhilesh Gupta, Senior Advisor, DST. Then Dr. Ranjan Bhattacharya, Principal Scientist, IERI, New Delhi. Dr. Ranjan Bhattacharya ji, Principal Scientist, IERI, New Delhi. Professor C. Rajasekaran, VIT, Vellore. Professor C. Rajasekaran, VIT, Vellore. Then Professor Atul, CHKPAV, Palampur, are their representative. Okay, come, madam. Then Dr. C. Biradar, C4, ICAR-CAFRI India Officer, New Delhi. These are the fellowship from the society. Badaiyo, sir. Okay. Now, Dr. Saurabh Sonwani. Saurabh Sonwani, please. Now there will be exchange of documents of MOUs signed between ICAR-CAFRI Jhansi and World Resources Institute India. So I would like to representative from World Resources India and our CAFRI. हाँ सब MOUs between ICAR-CAFRI Jhansi and World Resources Institute India. The exchange of documents will take place. Thank you, sir. Thank you all.
Okay, so it's not the end. This is only to encourage the people to keep up the spirit of doing research, doing science, doing innovation, and grow together mechanism, all for Neem and in the name of Neem. And this Society for Science of Climate Change is also committed for the promotional activities of Neem together. And in the end of this felicitation and award ceremony, I would like to extend a wholehearted welcome and thanks to our Honorable DDG. अरे भाई क्या तू रखो ना इधर. So we want to extend heartfelt thanks to our Honorable DDG Dr. S.K. Chaudhary, who has been a guiding force for all of us in the sphere of research on natural resources management, being the SMD head, subject matter divisional head of the natural resource management subject in the Indian Council of Agriculture Research. He has been involved in various scientific, technical, advisory and policy making paradigms of the government of India with respect to climate change. So the Society for Science of Climate Change and the Sustainable Environment deeply regards the services of Dr. S.K. Chaudhary and would like to confer in the witness of all of you and Neem the honorary fellowship award of the year to Dr. S.K. Chaudhary. So I would like to request Professor R.P. Singh to felicitate Dr. S.K. Chaudhary.
Thank you, sir.
Oh, okay, ठीक है. Now, with this wonderful series of events for the last 2 days, we have been specially witnessing the whole protocol and paradigm with regard to Neem research development, tools and technologies, products and also operational chemicals that could be possibly used for sustainable agriculture in this country has been showcased, and we are all witness to that, and Neem in particular is also a witness to that. With this, we request our Honorable Chief Guest of today's valedictory session, Dr. S.K. Chaudhary, who is the Deputy Director General of Natural Resource Management of the Indian Council of Agriculture Research, for his Chief Guest address. Please. Oh.
Thank you so much. Namaste to all of you. Good evening. First, let me congratulate the World Neem Organization for organizing this very, very important event, which is very rare in the name of a tree. And therefore, it is very unique, and that too a tree which has great importance in India, Indian literature, and Indian society. A tree which is considered to be
a very, very important tree in all the dimensions and all the cross sections of life in India. And for that, I would like to congratulate the organizers who did a lot of efforts in organizing this conference. On the dais, Dr. Gupta, Dr. R.P. Singh, my colleague, Director of CAFRI, Dr. Arunachalam, Nisha Mendiratta ji, Dr. Balaji, Dr. Vyas, and Dr. Nirmala, who did consistent efforts. In fact, I wanted to be present in the inaugural ceremony of this function, but because of some other urgent assignments outside Delhi, I was not able to be present here today. But I did not want to miss this. Although today's schedule was also very tight, but still we squeezed some time for this important event. Dr. Vyasji, when first time he talked to me over phone, I was not able to take it very seriously. I was thinking, who is this gentleman who is talking about Neem, and really somebody is there who seriously wants to have an event on exclusively Neem? Then one day he came, we had discussion across the table, and then I could feel that there is a great sense of seriousness. I immediately called Arunachalam, my friend in Jhansi, and he said, yes, this is very important organization, and we have to extend our support for this. And immediately ICR agreed to be part of this important event. And I'm thankful to you for bringing so many people here from different areas, different arenas. Otherwise, our conferences are most related to agriculture and agriculture uses. We are not going beyond. In Vedic literature, it is mentioned that Phalena parichayate vriksham. Phalena parichayate vriksham. If you want to taste any tree, just observe its seed. And from that account, if we talk of neem seeds, people may take it positively or negatively. But tree is so important, it has corrected the Vedic statement of Phalena parichayate vriksham. Now, because the root systems, the overall importance to the society, and most importantly, its role in cleaning the environment, providing high quality ecosystem services. Although all vegetation provides ecosystem services or supports soil to complete the functions in terms of ecosystem functions or ecosystem services, but special mention to Neem because it enhances soil's capability and capacity to really function positively towards the ecosystem services. And therefore, this single function itself, it justifies why Neem is given maximum importance in this country. When we say that Annad Bhavanti Bhutani Parjanyad Danna Sambhavaha. Annad Bhavanti Bhutani, if there is no grain, there cannot be existence of living being or human being. At the same time, Parjanyadam Anna Sambhavaha. And this grain production is possible because there is a rainfall or there is precipitation. And precipitation has direct link with the trees grown over a given area in any country. Concerns are raised, Government of India very seriously trying all the state governments and their forest departments to enhance the forest cover all across the country. But other than the forest cover, if you are to talk of the trees outside the forest or trees which have directly entered in the hearts of the society or social matrix, it is Neem, which stands on the top of all the trees. Therefore, special programs on Neem and research programs on Neem, this is very, very important. With all the development and the rattle race of development, we cannot forget trees like Neem. Of course, Ashwath Vriksha, it has relevance in Indian philosophy and Indian mythology, but Ashwatha Vriksha may be having importance for a specific purpose. But Neem is a multidimensional, multi-purpose tree which does not require any introduction or any kind of special emphasis in any society. When I was doing my post-graduation those days, Indian agricultural scenario, particularly the soil science and soil physics community, we were busy in finding out something which inhibits the nitrification of urea. When urea is applied, nitrification or immediately hydrolysis happens. All your scientists, you know very well. After hydrolysis, immediately nitrification happens. Some enzymes are responsible for that, and some bacteria are responsible for that. If the process of this nitrification is arrested, then probably it will enhance the use efficiency of Neem. This kind of understanding was there. With that backdrop, researches were going on. The area where you are sitting today, Indian Agricultural Research Institute, here also the researches on Neem coating over urea started long back in 65s and 70s. And those researches led to a perfect technology in the name of Neem coated urea, which is being sold today all across the country. Dr. R.B. Sinha, our senior colleague who is sitting here, that time he was Joint Secretary in Government of India, Department of Agriculture. And those days, we were trying to standardize how this neem oil or neem extract will be perfectly fitting and how to draw the quality controls. Because neem grown in Tamil Nadu, neem grown in northern part of India, neem grown in western and eastern part of India has altogether different compositions so far neem extract or neem oil is concerned. Then we were very much worried how to do it. Then ultimately we found out some way out, and ultimately we came with the three ingredients which are very well known, and some of them they were discussed at length. Some presentations I saw, they were there. So nimbin, azadirachtin, and sapolinitin or something, these three contents are there, salinine. So these three contents and their individual individual components concentration and overall concentration we standardized in the fertilizer control order. Then these oils were allowed to be coated on the urea. And not only this, there are so many things, so many uses of neem which are directly rooted in the society. In Maharashtra, some people must be there present in Maharashtra. When newborn is there, बच्चा जब जन्म लेता है, so a kind of litter is placed with green leaves of neem, and that child is allowed to be there on that litter for hours together, so that the initial jaundice and pneumonia, these two problems can be avoided. Likewise, every one of you know the n number of uses of neem. I am very happy that during this 2 days deliberation, effectively half day yesterday and half day today, you discuss almost all the points which are very, very relevant for neem. And the wonder tree, if you see all across the countries, I have one observation. This is not only the social uses, medicinal uses, or ornamental uses or chemical uses, neem has also got a different dimension. You also think of this. After 1400, or when before the period where there were many invention on Indian community, or after that, I do not want to name a specific period, but after 1400 or 1500 year, means 1400 या 1500 के बाद, if you see the people whom we call saints, or those who were limiting themselves in caves or forests, in and around their habitats, you will find this tree, neem. Besides all external and ornamental uses, this is the best possible internal use of neem. what we call under the Neem tree, saints, they performed their tapasyas. They performed their sadhanas. So this tree has a different meaning which is not understood very well. If you just see the old photographs of those saints, you will find there is a Neem tree. Those who are visiting Shirdi, you will find there is a reference of Neem tree. Those who are going in the Arunachalam hills of Maharshi Ramana, you will find a reference of Neem. Those will go to ashramas of Osho's, Rajneesh, you will find a definite reference to tree. And many, what we call them Muslim fakirs, they also have this kind of trees in and around their habitats. So there is something which are not understood by us and which needs to be understood deeply. But for that, you cannot do this kind of research outside. You need to get first inside. Once you start getting inside, probably Neem tree will support you to reach to your inner self in a much better way. Probably that is what additional service being provided by this important and very, very important tree to the Indian community and Indian masses and societies. Friends, I would like to congratulate all those who have received awards working in these fields, including the climate change, and also to the office bearers who were involved in arranging this very important conference of 2 days. You have traveled all across distances and you listened all the deliberations very carefully. My colleagues from ICR, they are also here, and also colleagues from DST and other organizations, they are sitting over here. And those who are directly or indirectly related to Neem, my best compliments to all of you. Keep doing wonderful work in the area of Neem. Our dedicated institute, which is located at Jhansi, Central Agroforestry Research Institute, has a huge and large number of accessions of Neem. If you go there, you will really enjoy moving around those Neem accessions. And they are very well conserved, and they are available for uses, for research purposes. And also, if you have some use of those accessions, definitely you can contact our institute at Jhansi. The institute is very, very important institute in the area of agroforestry and providing very good quality, high quality planting material. Not only Neem, in area of all important agroforestry tree species, but more specific to Neem, our institute offers so many services there, and we welcome any suggestion and also your ideas and any new innovation. We would love to work with each one of you if you wish so. So with this, I would like to stop here. My best compliments and thanks to the organizers for inviting me in this valedictory session of important conference. Thank you so much. Namaste. Jai Hind.
Yeah, thank you so much for those wonderful words that came from the Honorable Chief Guest of today's valedictory session, Dr. S.K. Chaudhary. He pointed out from the societal commitments that we all must have while doing science on Neem was very much highlighted, and that's appreciated. Thank you so much for those golden words, sir. I also join the chief guest in congratulating all those awardees who received the awards today, and also in the inaugural session of a few awards were given. So all of them, the congratulations goes to all of them. Now it's my humble duty on behalf of the whole organizing team to propose the vote of thanks for this great, grand Neem Summit. First of all, I thank the Indian Council of Agricultural Research, the authorities, Dr. Himanshu Pathak, the DG of ICR, and Dr. S.K. Chowdhury, in particular, the Honorable DDG of NRM, who gave nod to establish this collaborative effort of holding this Neem summit in New Delhi. And with this, we also got this space for holding this very important event together. So thanks go to the organization, that is ICR. And of course, the summit has been organized with the support of various partners, right from the sponsors, the knowledge partners, and also institutional partners, the companies who came on board to put up their stalls and showcasing all the products which might have given more insights for the public to consume the knowledge and also take it forward through market and economy. So the thank you goes to each one of those individuals who are involved in this process of the trade fair that was made possible with the support of all of you. And then we bow down in front of all the delegates who participated in the Neem summit. It's a very narrow subject, so the number of participants might look very small, but the deliberations were quite useful, and implementable recommendations would emerge out of this summit, which will be submitted to the government in a week or so. And then we also profusely appreciate the collaboration, cooperation, and networking that we received from many individuals who also came forward to support and participate in this summit. Most importantly, the operational people on ground in the stage, in the audio visuals, in the electronic activities, the housekeeping, catering, all those people really are appreciated for their efforts, which made this summit seamlessly going through and timely closure as well. We also thank the individual participants who represented 10 different countries who all came on board for this summit. Friends, it is very important to say at this point in time, this is one summit, although it's a narrow subject, but still we could bring in people from at least 4 different ministries, from the Ministry of Agriculture, Ministry of Science and Technology, Ministry of Fertilizers, and also Niti Aayog. So people were there on board for this. So with this, we thank each one of you to remember Neem, to speak of Neem, to go with Neem memories, green memories, go back safe, say Neem before leaving. And I request all of you to quickly come to the dais so that we have a group photo and then we disperse. Thank you so much. Please come. Keep taking photographs. Yeah, please. Say Neem. Thank you.